Yeah, we want to be looking downstream.
Looks good. Gotta keep it. That's not the way. Okay, so now that is facing, so it should go.
Hola a todos, todas.
Looks like it wants to go that
Mi nombre es Paula Proaño Mesías. Soy la artista en residencia acá en Schmidt en esta expedición. Y les doy la bienvenida a mi proyecto de performance. Es una acción performática en la que se hace una ofrenda de gratitud y se pide permiso al océano y a las divinidades que representa con una pluma de ñandú. Este trabajo toma de la tradición lafkenche, parte de la tradición mapuche. Y la idea es tenerlos presentes en esta parte del océano y acá en la expedición haciendo uso de la tecnología de acá del SOI. Hello everyone, my name is Paula Proaño Mesías. I'm the artist at sea here at SOI, and I would like to introduce you to my project. This is like a performance activation in which we are giving an offering to the divinities that the ocean represents in Lafkenche tradition. So this feather is a rhea feather that holds a lot of meaning in this tradition. It is used for ritual making, and it also symbolizes pathway to divinity. So we're going to give this offering to the ocean to give thanks and to practice gratitude.
Let's ask Paula. Once we release, so what do you think of this lighting right now? Once we release it, do you want them to turn more lights on so we can see it, or do you want more lights on now, or different lights?
I like these lights now. We have the contrast, but after that we can change it. We can change the high low, so we can do that.
We should maybe, so we don't, because we might lose it very quickly. So you might want to put the settings now, if you want, before we release it.
Good.
Is that also?
I don't know if he- Yeah.
What's the focus on?
So I like
Have
the
fun.
contrast.
See you tomorrow.
Is our camera has a different setting or like that? Is just
This right here?
Yeah.
There's the two positions, so that we can yeah, it's gonna sit different now. And probably have the like different hierarchy.
This is the most accurate exposure.
Is it possible to change the one that's looking down just for this particular?
We can, it's just not being sent in.
Oh, it's not being saved.
No,
It's,
well, it's being safe.
yeah,
but
it's not being recorded in 4K. It's just in 1080.
Oh, okay.
I can.
I'm just advocating for colorless product.
But maybe for the recording, for Alex recording.
Oh, yeah, that's way better.
Alrighty, I'm ready to roll.
I'm gonna.
Is the camera all the way zoomed out?
Yeah, I think so.
And, I guess we just wanna watch it for as long as we can and when it goes away.
But,
The idea is to like. see
Nos
it
estamos
going away.
preparando para
so we'll watch
entregar
it.
la ofrenda
It's safe for the RV to do that.
y que esta se vaya con la
we
corriente.
can come back to it. If it's not going the direction we want it to.
Sometimes current is good, this is once that's good.
It should,
I
Yeah.
might be able to like
just kind of
come forward a
yeah,
little.
just watch it a little bit.
I think we
It's
wanna
like-
watch it go away,
Right.
like it's like an offering.
So if you can go like an out of frame into that direction, you can also.
Yeah, you can, I mean, let's see. Yeah, look where everything is going, so it's gonna go where all these sediments is going. And if it's going there too fast, like out there if you're too fast, I could always like rotate a little to bring it back in frame.
I think it's best to just open it on the table.
Yeah.
So, are we ready?
Yeah, I'm ready to do it. Okay. Whoa, I just died.
Oh, no.
Oh, hold on.
Take it off after that. You still have that arrow?
You think that one would safely? I don't know what it's like. Go back.
Nice.
Okay, we ready?
It's a squid.
See it.
Wow.
Yeah.
He's blessing.
Yeah. Oh, it's so good.
Try to get it off of there. Stick it there.
Maybe if you like back up, it'll
Yeah,
just
see,
kind of rip
Make it
off.
back up.
Yeah. Got it again?
Oh,
Which one's here? So we have two problems. For one, that stuff right there, the rest of the stuff, I don't know if I need to come back up to me. So that's where that is, I was gonna use 5 or 6, so I can, yep, this arms to this. Got it. Yep. You can use the other one, the other one is fine. Unfortunately, it's a very, very bad spot.
You want to try to switch controllers?
Sure. This isn't going on.
I'll switch.
do you see it making those flashing? Is it supposed to do that?
Just needs to step power.
Ah, I see.
Same thing?
I just don't know which one I have right now.
Yeah. That one.
There it again. Go down. What's that?
Looks like the lower clock.
Yeah, so, alright, I mean, can do whatever it is. There it goes. That took a lot longer than normal.
Kind of swing outboard.
Hold on. It failed again.
I think it might be touching the desampler lid.
Yeah, but I can't control it to move it off of there. That's it.
Yeah.
inclusion. You submit yours. Shred.
Just tie the feather to the.
It looks like there's a string around it.
Yeah, we had to tie those shut in case it did not, like it was doing before, it was falling off.
Oh.
And the feathers caught in the floor. Less than instructed.
Okay.
We're gonna shove this, leave this out. Alright. This on does not have the same problems as that on. So it should not be that.
What depth do you want to do the other ones at?
Not 500.
Okay.
And that could be like a nice experience.
Yeah.
Slowly unload.
Yeah.
Okay. So,
500.
no. Well, are you gonna do it later, or you want to go straight down to 500 and do it?
I can do
Like,
it
what's the
later.
plan?
So you can start with
With doing the transects? Okay.
is it okay that I come like in the middle of the transit, or you prefer that we do it now?
I'll say.
No, as long as it's
Yeah.
okay with you that we do these, I guess, how many down to 500? 1, 2, 3.
Three 15-minute transects, and then we'll get to 500, and then we could do at least that one.
For me, it's okay.
Is that fine?
Yeah, it's fine.
Okay. Then let's just do that. Okay, so then we're gonna,
Yeah.
We will have to change the lighting for your needs, but for me, it's okay.
yeah, that's
Yeah,
fine.
I don't mind.
It's not too hard.
So we'll do the first transect at this depth, and then we'll work our way down to 500 and then release the other two.
Okay.
So we just need to change the lighting, and let's test out doing it how we had it last night and see if that looks good, or yesterday, which was a ISO of 320.
Do you know what the?
Yeah, all the lights were on. The ISO was at 320, and it was the iris was 2 clicks up, two clicks up from
I
the
think
post.
that's better than we can do.
And then autofocus is turned off, and it's turned to a midpoint focus. And I think that's it.
Yeah, I think that's worth it.
And then is the focus like turned somewhere in the middle?
Yeah.
Yeah. And then we want to face into the current, if possible.
More or less.
Yeah, it's a
And are all the lights on forward?
That is all looks like the center of the
Okay. Yeah. Yeah, we were just trying to get it as evenly illuminated as possible. That looks pretty good.
I think that looks pretty good. What do you think, Lee?
Yeah, are they?
This is the settings from yesterday.
The camera settings. Yeah, I think that
Yeah.
looks the same.
Okay. All right. Well then, I guess.
Lee, maybe you can show me how to log too.
Yeah,
So-
it's really easy.
Yeah. So then, oh, this is so beautiful.
There's so much stuff in the water.
So then we just drive forward at like 0.2 meters per second.
And you wanted that to 125?
125.
So 0.2?
0.2.
Bridge RV.
So you just hit
Yeah,
the
so
add
we're
button,
gonna
or
start our
I don't want to
transect.
make it
We're gonna
one.
be
Mobile
going
version.
current, so we'll be starting to forward. We're going at 0.2 meters per second. So we're gonna start going, and we'll get to about midships. That's where we're gonna want to stay.
There we go.
Oh, maybe it was.
So
Yeah,
Oh no, that's just adding a.
tabs.
hello.
This is like reviewing.
I don't
All right,
know.
can you turn your ROV off or something? Because that's really bad.
Okay.
I only have one thing.
Maybe one of them can show us how to
All
work.
right,
I don't
that
know.
was just the P bridge. So yeah. All right. Yep. You got me. All right.
Maybe Josh.
If you can monitor PROV only, please.
Yeah, I am on the ROV.
Okay. So you can hear me? Can you hear me?
On monitoring on the ROV.
Can you hear me now? All right. So it's not, you're not hearing me on PROV, so I'm going to continue on P bridge for you.
Do you have an account? Or should I log in?
Oh, I don't know
Yeah,
what
so
my.
we're gonna be
I think
going
I
at
made an
0.2
account,
meters
but I have no idea what my
per
login
second.
is.
Straight ahead.
Okay.
Okay, 0.2 meters a second copy on sorry.
All right, thank you. All righty. Happy?
Yeah. I've started.
Okay. Yeah, this looks great. All right. I'm gonna start the timer. I think it's fine if. Oh, are you starting the? You figured out the logging? That's. I'll start the timer. You can just log it whenever you figure it out. It's okay. Ooh. That was pretty. Yeah, the water looks totally different from the other day. It's like a lot more goopy.
ROV bridge, that's me start making the way now.
Thank you very much.
Lee, do you know what these things are? They're gone.
Is the speed good?
I think so. Well, you could go a little faster, like 0.2, 0.2 to 0.3. Yeah. Wow. Yeah, I think that's good.
I have it on my account.
Okay.
So you just do video transect, start, and then whatever the target depth is.
Okay.
And then maybe just make a note that
125
this observation is late.
meters. Okay.
Well, okay.
What?
That's okay. I was just saying you could make a note that it was started late, but you think you can add a comment. Yeah.
Okay. Okay.
Or that it just that
Logged.
the log is like.
Oops. Oof.
There's this
Logged.
out.
Logged. Let's see. 2.5 minutes. Oh wait, 2. And 2.
And then for the end of it, it's the same thing. It's just there's you hit the stop instead of start.
Oof, I don't even know what you pushed.
Well, I'll be here, but yeah, you just hit video transect, and once you hit it, don't do that yet. That'll be like logging it,
Oh, okay.
and then you can select.
Oh,
There's
okay.
options for it to be the start or the stop.
Oh, okay, cool. All right, that
It's
doesn't
very
seem too
simple.
hard. Okay.
I think you can figure it out.
All right.
Okay, so, alright, then at 9 o'clock I'll come relieve, but the thing is,
And then
now
there's,
you can now, maybe 2 people.
there's also buttons for like biology sample, and
Oh, okay.
it's similar. You just like fill out some things, but my understanding is once you hit the button is when it logs it,
Okay.
and then you have time to like fill it out.
Okay.
So it'll like ask you what jar it went in and stuff like that.
Okay. All right. Thank you. Ooh, nice. This is really good.
Yeah, you could come in at 9.
Lee, do you know what the strings are with the blobs?
I think they siphonophores.
Siphonophores?
And then I've seen a bunch of those.
Oh.
Leptocephalus seal larvae.
What do
Those
they
are,
look
the
like?
things that like swim away. They're kind of transparent.
Oh.
They have these really tiny little heads. They're so funny looking. I bet they caught some in the net if we're seeing this many.
Yeah, I saw a lot of that.
And then I've also seen a few Furrowy ctenophores.
Cool.
This is really
Yeah,
cool.
this.
They flew?
We were flying hard.
Yeah.
Oh, yeah, so that's the eel larvae that just swim away.
Oh, I see.
There's so many of them now.
Ooh, yeah.
Oh, that was one of those long ctenophores that we saw from the end of the
Oh,
last
I
night.
saw that one. Yeah.
Yeah. But a lot of these aren't migrating.
This footage is really beautiful. Like the ROV isn't going up and down. Like there was on some other days the ROV was kind of like doing this thing through the water, but today today it's perfect.
Seems like there's less current.
Yes,
That's the pilot's totally fine.
Nice job.
But yeah, there is some, to be honest, there is some, when the current's pulling, it makes it a little bit more difficult.
No, yeah.
But it's mostly cloth. But it's mostly cloth.
You guys don't use like an auto altitude? You just do it manually?
Auto altitude wouldn't work until we get to get a return on it, and it's only about 5 meters where it's
okay.
consistent. Yeah. We can do an auto depth, which uses a pressure sensor and some other things, but the issues we have with that, especially in windier conditions, is that the water above the ROV is constantly fluctuating, and it's pretty sensitive, so it's gonna total it, but get rid of it.
So it's better just do manual.
Yeah. But the idea is that we try to trim the ROV so it's as stood behind you. It's as neutral as possible. So instead of down there, it doesn't want to.
I was like, how is that where we are? Like, no, not yet.
Where we're going. She wants us to do an RV
Do you know,
out there.
what's going on with the geology there?
No.
Well, we find out.
Ooh, that looks like a
Are they dying to go to the
formation,
seafloor?
maybe.
I mean, I think it'd be fun to do an RV out there.
I agree.
It's a place where we can get to the bottom and it's within our depth of where we wanted to go anyway.
Well, and you can have a cool concentrating effect with the DVM.
Yeah, I was wondering if it might
Yeah.
affect things, how the fish might respond and everything.
Ooh.
Yeah, because you
Nice.
tend to see like
Very important.
you'll get
Interaction with the pelagic.
yeah, you'll get like pelagic fish right next to the
Yeah.
seafloor because they get compressed.
I know, it's like bluntly asking the very thing and not looking.
I'm so saying this, and then if we don't do it, we'll be so sad.
Not that everybody's doing this, it's very important.
You just can't see it's fun.
Yeah.
You can get like vertical walls.
That's where it's usually happening, but I'm sure it's.
It's a community.
Timing the whole bit.
It hasn't been marked yet.
Oh,
No, all the ones that we put in, I know it's not.
That's true.
So if there's some tracks over there looking.
You're gonna come with
So
me.
probably some
12.
of the
That's
videos.
great. I'll check on you.
Yeah, so that's better on the port.
So it's not
this
on
one,
it's just like on
works.
It's not at the long station, it's on the way. It's a short station, so we can do it.
We change the masters
We changed it early.
and work.
I think it's in the additional control is
Application.
the board here. Well, not in the
I was gonna say.
in the can.
So we used to be
When
like
he shuts off, the slide shuts off. So something it's showing somewhere. So we're very short. It shows
I
the
saw
slide.
that.
The slide was
They
off.
start to blink.
Tell you what you want.
Same time it blinks the light for them. That's weird. Did you see?
Should have been
Did you guys exchange again? Is this controller on the left?
Yeah, virgin.
Well, but so this will be a recovery during the day. So when we're coming up, it'll be something we can cover. Right?
The arm should have crossed perhaps.
So it's not 12 hours.
Where is that?
Probably migrated before we start getting too far out. I don't know how to Midwater animals, but do you know?
see
the
through.
way that they light it is to show them, and then you can like turn that off and see what it's like actually.
Star volume in it, 24 volts, comes from the PSU12. Afterlight comes from PSU12, but nowhere here shows the char light. Probably comes from the 12 too, and they forgot to put it.
Maybe.
Is that us right now?
Yeah.
It's probably, yeah, well, no, 7. We're kind of between 6 and 7.
The fish.
I want to go down the middle of it. Obviously, a lot of material.
All right. Stop.
Doesn't show the char light on the list.
Oh man, it's like I've never used a computer before.
Did you figure it out?
Yeah, I think so. All right, so now the next depth is 250 meters.
So you ready to go down?
Yes.
Is Embari not use sea log?
No, Embari has its own
I don't know,
system.
where you are.
Just stop it as
What speed you will moving?
Huh?
You know the speed?
Just stop.
Bridge, ROV.
You're gonna start coming down.
ROV bridge, go ahead.
May you stop the move? We're gonna descend to 250.
Yeah, no problem. Okay.
Thank you. Hang on.
Hmm?
What's the maximum depth?
1500. Yeah. Josh, do you know if my microphone is on?
Yes.
Oh, okay. I didn't realize. My name is Colleen Durkin, and I am an oceanographer from the Monterey Bay Aquarium Research Institute. And we are here on the Animals as Living Bioreactors research cruise. We are a very interdisciplinary team of scientists and from lots of different countries. What we're doing today is we're starting out this ROV dive by surveying the animals in the Midwaters. So we are looking at depths between 125 meters and 1,500 meters. And at increasing depths, we are going to be just flying the ROV straight forward into the current for 15 minutes and taking video of everything we see. And then later we'll be able to analyze that video in order to identify and count all the animals that we go past, and we can normalize that to the area that we cover, so we can quantify what animals are in different depths in this part of the ocean. And this is just one way we can view the animal communities. Another way we can view animal communities and quantify them is through net tows off of the ship. And so that's another thing we're doing on this cruise, so we can view these animals, these animal communities in different ways. And so right now we just finished our 125 meter transect. We flew the ROV forward for 15 minutes. Now we're descending to 250 meters. And we're going to keep going down for the next few hours until we get down to 1,500 meters. After that, we're going to very slowly work our way back up to the surface, but focus on exploring the animals, looking at them in more detail, trying to find some that we might collect so that we can bring them back onto the ship and use them for our measurements. The types of measurements that we're doing on this cruise we're really focusing on how these animals affect carbon export in the ocean, so how the ocean's role in taking up carbon dioxide from the atmosphere and sequestering it into the deep ocean. And we know that these animals that live in the midwater and the mesopelagic play a big role in how much carbon actually gets transported from the surface into the deep. And one of the things that it's kind of our hypothesis of this cruise is that the microbiome, the microbes that live in their guts, are working together with the animals to transform carbon as it's sinking, and that the processes happening inside of the animal guts are really different than the processes happening in the seawater. And so whether carbon goes through an animal gut as it's sinking, all the particles, all this marine snow that we're seeing, whether that carbon goes through the gut of an animal will affect how much carbon actually gets into the deep sea. And this is kind of it's an idea I haven't really heard of too much before, thinking about both the animals and the microbes together, and kind of having this integrated view of how the ocean takes up carbon and the ocean's role in the carbon cycle. So it's exciting working with scientists from all these different backgrounds, and that's one of the reasons I was really excited to be part of this project. Oh, someone asked what animals we've caught. Lee, you can jump in here too, but they've caught some fishes, lots of these very abundant fishes that live in the deep sea called cyclothony and myctophids. Full disclosure, I am not an animal expert, so I've been learning a lot. Tons of little shrimps, which lots of different species of those amphipods, euphausids, krill, all types of different species, and we definitely have some experts on those on board. Gelatinous organisms like arrowworms or keating gnats, salps, other types of siphonophores, but the siphonophores and other gelatinous organisms sometimes get a little bit messed up in the net tows, and so that's one of the things the ROV is especially good at is observing and collecting these really delicate gelatinous organisms.
At
Looks like
250.
we're at 250 now, so we're gonna get started on our second transect. Gotta log it. Okay, so if you could just start going forward.
Bridge, ROV.
Yeah, go ahead, that'll be.
May you resume the move, please?
No problem, I'm like the man.
Thank you.
Okay, I'm gonna start our timer for 15 minutes. And let's see, this is the start of our 250 meter transect. Okay. And then on this dive, the other thing we're doing on this dive is our doing art performance with our artist at sea, Paula, and so she's here in the ROV control room. We started this dive doing one iteration of her art project, which she can tell us more about, and then we're going to repeat it once we get to 500 meters. So Paula, I know you explained your project at the very beginning, but maybe you could explain again.
Hello everyone, everyone that has been joining us and didn't see the first part. So I'm the artist at sea here in the expedition. My name is Paula Proaño Mesias. I'm from Quito, Ecuador, and here I'm proposing a performatic action within the ocean. So we did the first iteration when we started with the dive, and now we're going to do the second one when we get to 500 meters depth. Hola a todos, todas, los que nos están acompañando y que llegan ahora. Les cuento que en este dive estoy haciendo mi acción performática, la que propuse como proyecto para el Artist at Sea. Cuando comenzó la inmersión, hicimos la primera iteración de la performance, y ahora cuando lleguemos a los 500 metros vamos a hacer la segunda. El proyecto en concreto es un ritual de agradecimiento y de ofrenda a las divinidades que representa el mar en la cultura lafkenche. Lafkenche en español significa gente del mar, y es una comunidad parte de la comunidad mapuche que se encuentra en la latitud donde estamos nosotros, entre Chile y Argentina. Es una comunidad que trabaja mucho y lucha por defender su identidad territorial. Los pueden encontrar en internet como Identidad Territorial Lafkenche, y tanto en Chile como en Argentina han trabajado por conseguir leyes que les permitan cuidar y entenderse como parte del océano. Entre esas prácticas en las que ellos se reconocen como parte de esta red, de este sistema vivo, que es el mundo natural, ellos hacen rituales de ofrecimiento y para pedir permiso cuando necesitan pescar. Practican mucho la pesca artesanal. en ese sentido, a mí me interesaba proponer un ritual que haga eco a esas prácticas acá, en esta parte del océano, y como tratando de que tenga reverberancia, como estas intenciones más desde la espiritualidad ecológica, como una práctica que nos puede acercar a otros modos de vida más equilibrados, no tan antropocentristas. Así que para esta segunda iteración lo que vamos a hacer es dar una ofrenda de una pluma de ñandú. Les quiero decir cómo se traduce el ñandú en mapudungun. El nombre es choike, así que vamos a entregar una pluma de choike a la corriente a través del brazo mecánico del Subastian. El ñandú se utiliza mucho en este tipo de prácticas sagradas porque se considera que es un ave que puede acercarnos a lo divino, y es por esta razón que se utiliza mucho en la vestimenta para rituales, también en los tocados para el pelo. Ahora en esta ocasión la vamos a entregar a la corriente y van a poder ser parte de este momento con nosotros.
Okay.
Just like send me a message if there's anything I should know before I get up.
Sure, I think Anitra's gonna relieve me, and
Yeah.
then you'll relieve Anitra. Yeah.
I have a question for you, Polly.
Mm-hmm.
So, we talk about interested in organisms in the midwater.
Yeah.
How do you define midwater
Oof.
in the sense of, because we're like at 4,000 or 5,000 meter water depth, but we're then going to transit to a place where there's potentially a seamount that's 1,000 meters. So, would that midwater area change, or would it be a consistent depth?
I mean, so people usually think about it going down to like 1,000 meters, or at least, I mean, it kind of depends on which community of scientists you're talking to, and it depends on what tools they're using and also what questions they're asking. So if you're thinking about like long-term, like biogeochemistry, like when is that water going to see the surface again? Like when is it going to touch the atmosphere again? Usually people think like below 1,000 meters is long-term for carbon getting down there. That's long-term storage in the ocean because the currents that are that deep transport water, but it takes much longer for that water to come back up to the surface. Whereas the more shallow depths, if it's really shallow, it's either just going to get mixed up by a winter storm, and all that water will get mixed up, and so down to maybe, I don't know, 100 meters in a really bad storm, or depending on what part of the ocean you're in, or else the currents will move and reach a point, like hit land and come back up to the surface in only, tens of years or hundreds of years, not 1,000 years. So,
Okay.
so that's kind of I don't think I answered your question, but so when you get to a seamount, I think you would still call it the midwater, but it's kind of like hitting a seamount, and so then really interesting things happen at seamounts related to biology. So,
Okay. Yeah. I was just curious how you define, the midwater and when
Yeah.
it comes to, like, as you get shallower, does it change? Or if, like,
I don't
midwater
think
is
so.
defined
I
as
mean,
this, or-
I think that I'm sure someone else would have a different answer, but in my mind, the seamount kind of like rises up into the midwater, so it's kind of the currents that are moving are still going to be hitting that seamount, right? It's not like the upper ocean compresses. It's still, I don't think it changes.
Okay. I was just curious.
Yeah.
I just didn't know how it was potentially defined.
Someone might argue with me on
Yeah.
that.
That's how you define it.
That's my definition.
Okay.
Yeah, I think of it of like 100 meters to 1,000 meters.
Okay. Yeah, because I would be interested, because obviously Joan is trying to push for a seamount ROV descent.
Yeah.
And it'd be interesting to see the different diversity when it comes to it, because we are, doing the trawls at, what is it, trying to hit?
1,500.
Yeah.
That's why we're
So-
going down to 1,500 on the ROV dive. Right.
Yeah. So, which is also interesting, because then you have different sea life when there's a seamount, is obviously.
Right.
Yeah, so.
Right.
But which is why I initially asked, well, how do you define midwater? Can you define midwater when you're at a seamount?
I don't think it's defined by like
Okay.
distance from the seafloor,
Okay.
or I don't think of it that way.
I guess it's just what parameters you're looking at is considered.
Yeah, I think it totally depends on your field. And
Yeah.
like for people who study carbon export, it's like 100 meters to 1,000 meters.
I like it. It's very structured.
Yeah. But in the real world, like the ocean is structured differently. Like
Yeah.
like you said, there's seamounts that come and intersect the bottom. There's storms. Different parts of the ocean, like the Southern Ocean, and the North Atlantic, are going to be super stormy in the winter, and so the depth of the upper ocean that interacts with the atmosphere is also really different. So, people argue about this all the time. Really nerdy people. What's that guy? 30 seconds. All right, that's 15 minutes. Stop. The transect. 250. Okay. All right. So the next depth is 375.
Bridge, Arohi.
Arohi, bridge.
May you stop, please? We're gonna descend for 375.
No problem, stopping now.
Thank you.
So now we're going down to 375 meters to do a 15 minute transect, which just means flying the ROV straight into the current for 15 minutes at a relatively constant speed. And we use this to quantify the animals that we see in that straight line. The other thing we see a lot of are these white dots, all these particles, also called marine snow. And that's actually the main thing that my lab, my team at my Mbari study. So we have a lot of people on the ship who are experts in animals, and people who are experts in microbes and chemistry. And then my team is really focused on the marine snow, the particles. That includes the fecal pellets of all these animals. It includes fluffy aggregates, the byproducts of life, just sloppy eating, goopy stuff that falls off of animals. All of this churning of life leaves all this detritus in the water. And that detritus, the marine snow, it sinks, and that's all carbon, organic carbon and food that's sinking into the deep ocean. That's fueling all of the most of the life in most places in the deep ocean is fueled by that food that's raining down from above. And then, like we were just talking about, those sinking particles in marine snow also take carbon away from the atmosphere. It's being transported deep into the ocean where it's not going to see the atmosphere for a very, very long time. And that's one of the major ways that the ocean regulates carbon on the earth, takes up carbon, and regulates our climate. And so we're really trying to understand the biological processes that control how much carbon, how much marine snow sinks into the deep ocean. And it's obviously, we can see, regulated by really complicated biology. And so that's one of the things we're trying to do on this cruise is to try to, open up the black box of the biology that's happening in the midwater so we have a better ability to predict how much carbon might be getting into the deep sea. Or at least that's my motivation for being on this cruise. We have a lot of a really diverse group of scientists here who are all we're all motivated by that big picture idea, but people are passionate about a lot of different things. Yeah, somebody's asking if the ocean regulates carbon like forests, and yeah, it's like forests, except way more. The ocean plays a much bigger role in taking up carbon than forests, because it just has a huge capacity for taking up carbon compared to land, compared to forests. The amount of carbon held in ocean water is by far the largest carbon reservoir on Earth. And so yeah, it's kind of like forests, but you know, obviously I'm biased, but it's like way more important. I think a similar thing about forests is people think a lot about soils in the forest and the microbes, like the fungus and all the bacteria and things that live in the soil, and how that controls how much carbon can be taken up in the soil. And that's kind of like, you can think about that in the ocean as well. All of the microbial life and the animal life in the seawater is also affecting how much carbon the ocean can take up. And people are asking about the feather offering, and we're going to do it again at 500 meters. Right now we're going to 375, so after this we'll go to 500, then we'll repeat that, the art performance. Yeah, it's okay. 375. All right. Set the timer.
Bridge ROV.
Hello, the bridge.
May you resume the move, please?
No problem.
Thank you.
Okay. I'm going to start the timer. This is the 375. Okay.
Hello, the bridge, that's on the move.
Copy that, Bridge.
All right, somebody is asking, do I work in Monterey? Yeah, I work at the Monterey Bay Aquarium Research Institute, which actually is in Moss Landing, which is on Monterey Bay. It's about 25-minute drive from Monterey. It's right in the middle of Monterey Bay, where the deep canyon there comes right up to Moss Landing. Paula. Somebody, sorry, I don't speak Spanish, but Paula speaks Spanish, and also someone's asking about where you want the feather to go. They were wondering if you put a weight on it or what.
When we're going to do the second frame.
I don't think she understands your question.
Oh yeah. Somebody asking.
When are we going to do
No. How have you? They want to know. Are you putting a weight on the feather? Will it be
Oh.
left to float neutrally? Where will it go?
the feather?
Yeah.
Will go into the current and then disintegrate at some moment in time.
Yeah. There's no weight on it.
No, there's no weight.
It probably just floats away.
Yeah.
It will. I bet it will get eaten.
Do you think something is going
Oh,
to
yeah.
come to you?
Oh, definitely.
It's pretty big.
It will get eaten.
It's
But
like,
at least by bacteria.
okay, yeah. Yeah, maybe.
Amphipods. They love eating everything.
Really?
Yeah, it'll get eaten.
Okay, yeah. I
For
think
sure.
that's best.
It's like fresh food.
It goes into one cycle.
Yeah, it's gonna get turned into marine snow.
That's just for full circle with
Yeah.
the whole expedition.
Yeah. It's definitely gonna be eaten. There's no way you could. I don't think you could put anything in the ocean and it won't get eaten.
Yeah, it's not plastic. It's bio.
Yeah.
So
Even if it was plastic, it would get eaten.
you're probably right.
Yeah. Ooh, nice. Ooh. Yeah, the ROV looks so good today. It must be like the current isn't quite as strong, right? Like it's not, well,
I
it's
already
it's
told you
you.
what it is.
I know.
It's the
I know
It looks
it's
so
hard to
good.
believe, but
I know. It's not hard to believe. Ooh, what do you think that is? No stopping. Fish? No. Probably a fish, right?
yeah, that's like a fish.
Yeah. 7 more minutes. Oh, someone's asking what's causing such high oxygen saturation. I'm looking at that, and it's actually not saturated. It's like 78%. I don't know exactly how that sensor is like calculating that, but usually it's related to like how much oxygen is in the water, the temperature, and the temperature that it's at, and then there's just basic chemistry of how much oxygen can dissolve in seawater, and so there's the theoretical how much oxygen should be in the seawater, and then the measurement of how much is in the seawater. And so if it's less than 100% saturated, that means it's been breathed. The oxygen that's there has since it was last in contact with the atmosphere, when it was at the surface, organisms in that water have been breathing and using the oxygen, and so that's why it's less than 100. And as there's some depths in the ocean where it actually gets really, really low. There's some parts of the ocean where there's almost no oxygen, and the organisms that live in those depths have to have special adaptations, and there's really interesting chemistry that happens in those depths. But once you get into the deeper ocean, the only way for the seawater to become oxygenated again is ocean circulation, like physics. When that deep water finally hits land and upwells, or a very big storm somehow mixes that water back up to the atmosphere, and then it can become oxygenated from the atmosphere. That's also when carbon dioxide that's in the deep ocean gets aerated back out into the atmosphere as well. So when things are using oxygen, they're breathing out carbon dioxide, right? So as you get deeper in the ocean, there's less oxygen and more carbon dioxide, and that carbon dioxide also is just going to be stuck in that deep water until it circulates and interacts with the atmosphere again, and then it outgasses into the atmosphere. So that's why it matters how deep these carbon particles sink, because The deeper they sink, the longer it's going to take for that deep water to be mixed back up, or the ocean circulation to push it back up to the atmosphere. Sorry, someone asked about oxygen, and I'll just talk about carbon. Sorry, I don't speak Spanish. All of our, many of the scientists on the ship who speak Spanish are processing samples right now. We had a net come up, a mock nest net come up right before this ROV dive, and most of the scientists on the ship are right now in the lab processing those samples. It's really intensive. They have to catalog every single animal, weigh them, they sort them. It's really beautiful, actually. They're in all these trays, and every tray has the same type of animal. I think our media specialist on the cruise, Alex, is making some videos, and so you should be able to see what that looks like. It's really impressive, the amount of work. I mean, there's a huge amount of animals that come in those nets, and they catalog everything, and then after it's cataloged, different teams take different animals for different purposes. Mostly we're focusing on gut contents, but there's also a lot of taxonomy, genomics sequencing for animal evolution studies. So all the samples are being fully utilized, but it takes a lot of work. And unfortunately, most of the Spanish speakers on the ship are in there. Oh, there we go. Stop. That was the 375. Transect. And so now we'll go down to 500 and we'll do Paula's art performance.
Bridge, ROV. May you stop the move, please? We're going down.
Copy that. Stopping the move.
Rock back.
Feel the migraine. I don't blame you.
Just to keep like everything warm. It would be like really similar to that.
Hopefully not.
Yeah, hopefully that part it doesn't get stuck, but the idea would be have it here, like in a good position, just like euro. We could stay like that for 20 seconds until we get in position for loading, and we have to open the method. And Alex is going to
Yeah.
Big guns.
Right?
Yeah. Try it.
Well, getting there. Oh, I forgot about my tea.
Hey, you want some mirrors before you get on the camera?
Are we using the feather right away, or we're gonna do the transect first?
No, yeah, let's do the feather first.
Feather first.
Yeah.
Yeah, you get 2.7 turns.
Hmm?
You have 2.7 turns in the vehicle.
That has to be big.
I'm gonna check with that thing out.
I happen to change headache.
Yeah,
Since
when
I've got it.
you get the thought,
Yeah.
just go straight away and then return it. So we just
So I guess we want to be facing away from the current.
Away, down,
So looking downstream of the current, if that's possible.
I did it for you. Yeah, nice and bright. No, it's okay. No. Okay. Yeah.
Whenever, so maybe we could, are we facing downstream now or?
Yeah.
I mean, it seems like the particles are coming toward us, right? I think we want everything to be flowing away from us.
If you look at the currents, they should be going from the northeast to the southwest. So it should be, I don't know why. Yeah.
That's as we trust. So it's probably pulling.
Oh, okay. Well, then
Trust.
let's just wait. If we just wait for it to calm down, or
So you turn your auto heading off and see if it fills out.
Yeah, 1 second. Let me just come back up to 5.
Right now, you just set up for it to be going this way.
Yes. Very still. But
okay, that's fine. As long as it's not gonna like flow
Come.
right back into
Yeah.
the camera, we want to see it flow
yeah,
away.
I think we are in correct.
Okay, and then maybe, can you turn on the, is it on the autofocus now?
Yes, on the specs you ask for lights, and the specs you ask for the cameras.
From last time?
Yes.
Okay, so maybe we could, if you hold the arm out, let's see what the lighting looks like and see if that still looks good here. Ooh, the squid loves it. Another squid. And then maybe, could you move the camera to get the samplers out of the view?
Yeah.
Yeah, he's just doing this so it's safely to move the arm right now.
Yeah.
Hold it in front of us. Just watch your bottom of the airborne. Actually, it's
What do you think of this lighting?
I can change position for you, Paula.
It could be more dark.
Close your eyes.
That's good.
You want to zoom or you want a picture like this?
Paula is the director
You wanna
now.
zoom like this?
Yeah, yeah.
Open up, really.
So it's starting like in the joint.
Only one to open power.
I think that's looking good.
Open up.
Yeah, open.
So we can move behind on it.
It's gone.
Move behind it.
Of course, there's no current in that water.
Okay, create the current.
It's
The
going to take
thruster.
some time.
Because we'll come back to check the pressure for that. That went much smoother than the first one. So, that was tight. I didn't know
Cool.
I mean, we could,
Well,
Find the
we
clearer
could,
for you.
even expect that.
how lucky are you? Believe.
Yeah.
You would start to the transect.
Are we done? Okay. So we just need to adjust the lights again. So back to manual focus, ISO 320. 2 clicks up from the closed iris and all the lights on.
With 2 clicks down to close.
Yeah, so if you close it and then go up 2 clicks.
So Chris, should I reset it at this
So go, yep, straight
this
off.
angle
Yeah.
and reset it there now?
Yeah, yeah.
You turn it off down to almost right.
Yeah, I should.
What lights were on before? Mid,
All of them.
get.
All the front ones, yeah, uppers, outers.
Ooh.
Yeah.
Turn up.
Upper, yeah. You guys good in there?
Center
Copy.
cat was on.
Yeah, you can turn it on. You guys see anything? Turn it on.
Perfect.
Set up the electric bill.
So, I think something's off with the settings. It's not as bright. Is the ISO up to 320?
ISO is good.
The iris.
When we did it the last time, I put it down to, I closed it and I went up 2 and you couldn't see anything, so I just kept going up until it was right.
Oh, okay.
Yeah,
Huh.
'cause I think what they probably did was turn auto off and then went up to turning auto off.
Oh, okay.
That's the only thing that would make sense to me.
We want to
Okay,
turn
maybe come it
the
down,
on
come
and
down a
off.
little. I think that's a little too bright.
Colin, do you want to try turn on and off just to see where the automatic goes?
Yeah.
No,
I would try it. Just turn auto off, on and back off, and then go up 2 and see if that's what they meant.
I
No,
think I can.
That is auto, and now I can turn off when it stays like that.
What ISO is it on?
320.
It's 320.
Yeah, sure, I didn't touch it. Talk about that.
So maybe turn the iris down 1.
Down.
Yeah.
One. Close one.
And is the focus somewhere in the middle?
Yep.
Could you go a little bit slower, like
Just
0.3?
trying to
Oh, you're trying to
get
catch
my-
up.
Yeah.
Yeah.
just Kind of go through this one.
Yeah.
1 minute.
Yep. I'm just trying to. Maybe could you go down 1 click on the iris? I think that's better. Yeah. Well, can you go down 1 just so I can compare?
One down.
Yeah, down. Let's do that one. Do you know how much up from the bottom that is? Just so I can write it down and maybe be consistent. The iris, the clicks on the iris.
It doesn't have a measure on there.
Oh, okay.
Yeah. So I mean, you can go all the way down so you can't see anything.
Oh, I see.
And if you go up to, you still can see one.
Count the clicks.
All right, well, this looks good to me, I think. So, yeah, I think that's good. All right, so whenever you're ready to like go into the current,
I'm
let me
ready.
know. You ready? Okay, so when you start going, I can start the timer.
Bridge, ROV.
On the bridge.
May you please resume the move?
No problem.
Thank you.
All right. Start at 500. And I think you could go just a tiny bit slower. Yeah. That's better, I think. Like somewhere but sorry to be a backseat driver. Somewhere between like 0.2 and 0.3 I think is good.
That was Uber.
Hmm.
The other one, you want to break?
What do you do?
Huh?
What do you do?
Yeah. I'm working at 11. Okay.
It's funny boy. 10:30. Oh, hello.
Huh? 11?
Oh, you want to say?
That's another.
Guys, thank you so much.
Just using the trims to come up on them.
No, thank you.
How do you feel that went?
It was okay. We are able to do it one more time. So,
Yeah.
for the next time, I think it would be nice to have it for closer shots.
No tread.
Hmm?
No tread.
No tread?
Yeah,
No,
get rid of that.
No, I was just talking about that.
Yeah.
I think
And no
it's
starboard
better
arm.
now. It is going to
But the thing is,
be still holding. So,
we
Yeah.
thought they could
Yeah.
put it
and that is going to
right for
help
even in the
to
and come up on
be
my
a
shoulders.
bit
That's
more
just
smooth. Yeah, I think that would be more. So the shots is going to be because I have some microscopic shots
Yeah.
of some specimens. So I want to get some better.
Yeah.
So it would be another way to connect things.
Yeah. Yeah. I like how the animals were interacting with it. Did you
Yeah.
see the amphipod like swimming through it?
Yeah.
That was really neat.
Yeah, there were a lot of animals.
Yeah.
That's been really amazing.
Yeah.
The squid was like.
There were two squids that came up by it, two different ones.
Yeah.
Like the first one and the second one.
And I'm wondering if they are attracted to it or something.
I think they're attracted to the light of the ROV.
Oh, okay.
Yeah. Or that's my guess.
They are. Yeah.
Yeah, but they
Or
were
they're
cute.
just they were there to receive the offering.
Let's
To
hope
eat it.
so. They were hungry.
Yeah.
Yeah. I like that new dimension of the work that
Uh-huh.
it is being eaten.
It's definitely gonna be eaten by something.
And also like it connects with the whole expedition.
Yeah. Totally.
That's nice.
Yeah. All right. Well, we can do it again next time.
It would be like in the next station, right?
Well, hard probably, but who knows? Who knows what else will happen?
Okay. I
You know,
feel like
seems like we'll probably be at the next station. Yeah.
yeah. Okay. I feel like I'm prepared now.
Thanks, Corinne.
I just mean our schedule is kind of like
Oh.
our like when we do the nets and all of that is kind of always in flux. So
Yeah, you
It's-
have to flow with it.
yeah.
We definitely have to get 2 more dives in on this cruise though.
Oh, there will be. Yeah, yeah. I think it definitely more at the other like the other long station.
That's true.
900 is coming up.
Oh yeah, yeah, yeah. We have to get that. And it sounds like Anitra is kind of does want to do one at the seamount.
So I like that.
That's cool.
That should be a deck at 6 p.m. kind of a thing.
Oh, okay.
Or
Yeah.
2 p.m.
Well,
yeah, we can go like anywhere after noon to midnight as long as we get to fly around.
yeah.
We don't care about the other.
Well, I did hear Anitra
Like that.
saying it'd be like walking past the Grand Canyon and
Not looking at it.
not looking at it.
You say
delta at this depth, it's fine.
65, just
I mean, you probably get into 50, you know. We won't mess with it right now because we don't want to run the risk of messing with the
Good, very high.
yeah. So, what we've seen on these transects and we've collected on the different nets, is it consistent with what y'all expected in this area, or is it more or less, or different? Or?
Well, I guess I can just, I am not the expert on that, but what I've been hearing is that, this area is really understudied, so there were some thoughts about what might be found, but there's not really that many observations.
Okay.
So I think they had some guesses, but then they've been seeing other things.
Okay.
And not seeing things that they thought they would see. So, sorry, I don't even know. I can't even tell you.
Fair.
I'm sorry you're stuck with me in the ROV room. It's because I literally am the only one who does not need to be sorting animals.
Nice word.
Yeah, that was beautiful. But I think there's some, once you get to a certain depth, there's some types of categories of animals that you kind of see in most places, right? But with variation, and I'm not sure.
I would think just because of the amount of current that's in this area, you would see a lot of stuff because there's just so many opportunities for food and movement and whatnot.
Yeah.
Did I see lots of things in the first transect for the show?
I have no idea.
We did the first one, right?
No.
We did the first one?
Oh, tonight?
The shallows when you guys went down.
That was.
Claude.
That was
No.
You and Claude did that one, really? That was the first
one
level,
was
yeah.
that one?
There was more. I wrote down lots of siphonophores, a lot of those little wormy, clear, transparent, flat,
The ribbony thing?
ribbony
Yeah.
things. Lots of those, and then the particles were kind of bigger and more fluffy.
No fish.
There were some little fish. Yeah.
Because what I realized when we were more south, I was expecting much more fish.
Yeah.
Because of very rich water.
Yeah.
And we didn't see almost fish there. It's not cool. Maybe overfishing by the Chinese.
Yeah. I don't know. I'm not sure what you would expect.
Because also in Brazil when we are on the Santos Basin tunnel,
Mm-hmm.
parts of the
Yeah.
Mahi.
Uh-huh. It was a very open ocean where we were, though.
To answer the question, you know, what we're talking about is definitely one, or do we just see 1 second ago? I think that would find out what that was.
Okay.
Time is out.
Stop. 500. Okay. All right. So now we go down to 625.
Bridge, may you stop the move, please?
Stopping now.
Thank you.
Hmm. Somebody's asking if the ribbony flat things might be a gossamer worm, and
No.
I think gossamer worms, is that the same thing as Tomopteris? Is that a gossamer worm? That's the one with all the, we just saw one, right?
Yeah, that was what we just saw.
Yeah.
When it came along the
Yes,
surface
yeah,
side.
yeah. So I don't think it was it wasn't the gossamer worms up shallower. It didn't have a lot of appendages on the worm. It was like a flat,
A wide ribbon.
wide thing. Lee, who's a postdoc at Mbari, they thought they were eel larvae or yeah. So I'm not sure.
So what's that one?
What's that?
700 feet?
Yeah.
625.
625.
Oh yeah, what is that?
Genetic survey shows very
Oh, yeah,
restricted to
eel
Maori
populations.
population.
So Lee knows what they're talking about.
Maybe he's doing that.
It's crazy to see all the different levels of development in these pictures. Leptocephalus.
Are they larvae?
Yeah.
That's cool.
Leptocephalus, aka larval stage of Maori eel.
That's really, wow, why were there so many larvae? That's neat.
starts now just coming everything is on.
Uh-huh.
And then as it starts to develop,
Oh my gosh, that is creepy.
I would not go like that for you. I get too scared.
Well, hopefully you're not on shift when we get to that depth again.
Wouldn't get too much further with pretty far behind us.
Yeah, I will catch up later.
I would-
tried to do before, but I was like, I need to stop one function.
Yeah, I would definitely
Push
get
forward.
forward for a little bit though, yeah, just for safety. Just 'cause, I mean, being to the side is one thing, but being that far to the start. Big issue. It's easier with the ship and the drift, so they always come back with us, so we don't go so far away from them. So that doesn't matter where we start the yeah, from the drift, and then let them come back with us if we need to.
Someone's asking if we're collecting samples, and we will be collecting samples on the second half of this dive. First half is flying horizontal transects at increasing depths for 15 minutes, and that's for the quantitative survey. So this first part, we are not stopping. We're just trying to get video of all the animals, what type of animals, how many there are, their distribution at different depth levels. Once we get to 1,500 meters, then that's our deepest depth. And so we'll spend the rest of the dive slowly coming back up to the surface and searching for animals, looking at them, getting nice video of them, and trying to collect some of them, so that, and probably some of the more delicate ones that we can't really get in the nets, that the net tows that we're also doing on the ship. So that'll be later. Foraminifera at this depth. Are we just catching up with the ship, or yeah? Were you here the other day when they saw the Phaidarians, all the deep, those They like, and Itra said they look like COVID-19.
Yeah.
Yeah.
Yeah,
They're called phaeodarians. Yeah.
but the one that you got a little too close to when it couldn't.
I missed it. And Itra just showed me pictures of it later.
So we saw the one that we videoed for a while, and then on our way up, whenever we were trying to get back up to get it back up time,
Yeah.
we passed about 7 or 8 of those.
Oh, cool.
Yeah.
Oh my gosh, that's
Yeah.
really neat.
Like, I'd see them, I'm like, oh, there's that again, that's that again.
Oh, really?
Yeah.
Oh my gosh, I hope we see them today. What depth were they at?
I would say a little deeper than now.
Yeah,
Because
like
we kinda
deeper than
blew-
500. Yeah.
We blew past 1,000
Uh-huh.
and went to 500. And
Yeah.
I changed that with Doug. But they're
Cool.
is it them? Oh, sorry. But
Oh, that's neat.
yeah, so they're a little deeper. So I wouldn't probably push out.
All right. Well, I have my eyes peeled. We've been studying those in my team in Monterey Bay. They make their shells out of silica,
Yeah.
and they're kind of they have a very distant common ancestor with diatoms.
Okay.
So they're kind of we're looking at some of the traits, some of the genes that they share with diatoms for making those silica structures.
Okay.
Yeah. They're super beautiful.
Yeah, I'd never flown with it before.
And they're protists. It's incredible. They're single cells, and you can see them with the ROV. Okay, are we ready to transect at this depth, or just let me know?
2 minutes for adjust my delta. and
Okay.
we can- Yeah, you're ready. Tell me when.
Okay, let me get the timer. So once you start getting kind of like going into the current and it's like stuff is coming at us, then I'll,
Can you start any moment you tell?
you can start.
What
Pretty
is the
darling.
depth you want? 0.2?
Oh,
Tell me
can you do that move, please?
What's that?
Okay.
Thank you.
Oh, between the speed is between like 0.2 and 0.3.
0.2 and 0.2.
Yeah. All right, that looks good. Start. 625.
I saw that.
So your idea is you
What was that? Oh, yeah.
You
The
were-
Loretta? Yeah.
We even printed some, remember?
Yeah, we used a whole bunch of film that we were supposed to shoot. Yeah, that was definitely different. That was intense. It was only there was like 100,000 people watching. Okay. Yeah, the work that the scientists did was built like the science part of it. This is phenomenal. Daniel was like,
Still have one more. hopefully
Mm-hmm.
when he comes back to the TMS, to the docking station,
Mm-hmm.
why you come on?
Oh, a whale? When did that happen?
Years ago.
Oh, yeah.
I think you still have the video.
Oh, yeah, cool.
It's maybe 25, 20 years, no, 20 years ago
Uh-huh.
when I was starting.
Wow.
He was one of my instructors. That's my second year at
So that was
LV.
for 1 year when I got here.
That was 2007.
Uh-huh.
2 days before that.
Wow.
Colleen, how are you doing? What time of day is this particular for you?
Like midnight, I think?
Oh, okay, alright.
Or what time is it?
It's like 10.
Yeah. I can go a bit longer.
Okay. No, yeah, it's fine.
Yeah.
Whatever you wanna do.
Is this coming, we're at 8 a.m. is when this is coming back on deck? Yeah. That's fine.
His very first cruise, I think. He was so shy. Like, what he done for 6 weeks, probably. But then he got bored, he went to the because so yeah. And now it's like family now.
Very nice.
Do you know why at this step the ROVs, now the ROV's kind of like not doing this thing?
I
The
can try
reason is
demolish it.
because we're in auto depth. And auto depth is
Okay, because it wasn't doing that before, and now it started doing that.
the shitty smolder. Yeah. That's They had that. I think they had that. Years ago.
That was a larvation. There's a lot of larvations here, huh? At this depth. I'll write that down. Larvations.
Next to that, 750 volume.
Yeah, 750 is the next steps.
Have you been able to estimate the volume of water that we transit through for these?
What's that?
Have you been able to estimate the volume of water that we're going through for these?
Oh, I think I'll need to ask my colleagues how to do that.
Okay. So if you want, when we stop this transit, you can turn the lasers on and have that as a visual aid. It would be really hard to.
If you can think of a way to do that, I just don't know, like, it's not gonna, the lasers won't hit anything though, right?
But we'll still be able to like have them on there and see maybe the reflection of them on there, perhaps. And
If you have
get
a,
an measurement.
if you have an idea, then
Alright. That's
I'm
what we-
I'm all for trying it.
That's what I did with the Estefana from in Australia. Was I got the lasers on and we did an estimate.
Yeah.
Just what was in the thing and then just some simple measurement happened. So
Okay. Yeah. Let's do it.
so take that in.
Ooh. Wow. What's that? Maybe at the end of this transect we can.
When I go down, if you want, I don't know if you have idea how it works.
Sure.
See if you like water.
Okay.
I see
Yeah.
If you don't like, we turn off. If you like, we do
Okay.
lasers on.
Okay. That sounds good. So how much time we got? 1 minute. A lot of forams at this depth too. I'm gonna write that down. Forams. Keating maths. Cool.
Yep.
All right. Stop.
Where is our leak? And then I'll stop.
That was
Coming
6:25.
Okay. Can you try just to go into a drift for us a little bit and see if we can match it when we're going down so we don't get too far behind the vessel?
What do you mean?
Follow us. Okay.
Ooh,
You
nice.
barely see the lasers in there.
You have them on?
Yeah.
Ooh, I don't see them.
I can see them just ever so slightly.
Oh, I see them. I see one. Oh, I see them. I
Right
see
there,
them. Yeah,
right
yeah.
There, yeah.
Yeah.
The easiest way for me to get a real measurement is I would put the camera down and hit something on the porch.
Yeah, I mean, I guess the other thing is just if we see, we can see the D-samplers in the porch, so we kind of know, at least at that focal plane, that's how much distance it is. Right?
Yep. Would. Turn those off. Don't want to lose in your transects.
I don't want all ever, but we can ever.
Okay.
So I don't want this laser on ever, but I would hope that's.
The real world is dangerous.
Yeah.
Okay. Down to 750. And I am looking out for those Phaidarians. Ooh, cool. Sorry.
Right.
Hmm. All right. This is going to be a good depth. I can tell.
On your way.
It's going to be a magical depth. So you can go forward.
Can we start?
Yes.
Alright, Bree, we'll continue with that move, please.
Very good.
Thank you.
Okay. And I'll set the timer. Start, start, start. Okay. This is at 7:50. All right.
Is this speed okay for you? Points trooping
Yeah.
okay?
This looks great. Someone's asking, what's this dive about? So if you weren't here earlier to hear the explanation, this cruise is called Animals as Living Bioreactors, and so our team of scientists on this cruise is studying animals that live in the midwater. So generally animals that live between 100 meters and 1,500 meters deep, and many of these animals migrate up and down every day, come up to the surface at night, and then swim down deep during the day. And so what we're doing with our ROV dives is two things. First of all, we're doing these transects where the ROV flies horizontally for 15 minutes. And we're just getting quantitative videos of all the animals that live at this depth. So we successively move deeper and deeper. Right now we're at 750 meters. After this we have 4 more depths until we reach 1,500 meters. So this is our quantitative survey. once we reach 1,500 meters, then we're gonna take our time for quite a few hours, searching around, looking, trying to take a closer look at these animals, collecting some of them, and just slowly work our way back up to the surface, to try to collect some specimens, get some really great in situ or like underwater footage of the animals. Some of these animals we're also collecting using net tows on the ship. So, wow, this is a really beautiful siphonophore. Oh my goodness. Whoa. And so some of these are really delicate, like that guy right there. We aren't getting them intact in our net tows that we're doing. So the ROV is really great at being able to see what they look like in their natural condition, when they're alive, when they're floating in the water. Also can collect them more easily. Whoa, that is beautiful. And collect them intact. One of the things that we're measuring in these animals is their gut contents. So what's the microbial community, like the gut microbiome in the animals, and what's the taxonomy, how big are they, how many of them are there. So right now we're just driving forward, no stopping. That's why we ran over that siphonophore. Not allowed to stop. After we get to 1,500 meters, that's when we'll be stopping at different animals. Ctenophores. Ooh, what's that? Siphonophore. Beautiful. Somebody is asking how we collect animals. Well, when we collect the animals with the ROV, there are two ways that we can do that. And we can't move the camera now to look at that, but right below where the camera is looking, there are these jelly jars or detritus samplers. They're basically a cylinder with a top and a bottom lid. the top and the bottom lid move together. So when it's open, the whole cylinder is open, and when the lid's closed, they close together, so the whole thing's closed. So the ROV pilots fly the ROV so that the animal is gently goes inside of the cylinder, and then the doors close and it's trapped inside the cylinder. It's a really gentle way to collect animals, especially delicate animals like a ctenophore or other gelatinous things. There's also a suction sampler, so it's basically like a vacuum cleaner, and it sucks in water. And then there's some jars underneath the ROV that it gets suctioned into. We also on this cruise are using really large nets that are being towed for many, many hours behind the ship, really deep. So we can collect a lot of animals that way, especially fishes, more hardy animals like the crustaceans. There are some gelatinous animals that come up in there, and some of them are still like intact. A lot of the really delicate things just get destroyed in those nets because they're just being dragged through the water. But there's still quite a lot that comes up in the nets. It's a really good way to get a lot of specimens. This dive stream is going until we're recovering the ROV around 8 a.m. our time, so there's many more hours left. It is only, let's see, 10:30 p.m. our time, so we still have about 9.5 hours or 9 hours. Once we're done with these transects, I'm sure it will be much more exciting to watch, because that's when we'll be like zooming in on cool animals and trying to collect them. So this is the survey part. I will not be here for all 13 hours of this ROV dive. Yeah, the ROV pilots worked really hard on those samplers, and the lids close faster now. One of them also has a bottom, like a mesh bottom, so we're gonna try out to see if that works. Yeah, so we're also working on making the samplers work well. And that. The nets, so the nets that we are towing behind the ship collect some of the animals that we're seeing, for sure, especially the crustaceans, the fishes, and some of these jellies, especially like the salps, the arrow worms, those keating gnats. It's collecting a lot of the keating gnats, for sure. But the interesting thing is that using nets is one way to view the ocean, and using the ROV is another way to view the ocean. They both have a bias. Different things swim away from a net, and other things swim away from an ROV. So it's really interesting to use both tools. It's like a very complementary way of viewing the ocean. See where we're at here. 3 more minutes. the next steps is 875.
875.
Yeah.
125 increases.
Yeah.
The calibrating is the accompany which use it for those pushing sideways and moving the rest. Which compensates if the head of the babies need to be safe more just from side by side.
Yeah.
Now you have to go.
How deep are we gonna go?
Huh?
How deep are we gonna go from 4,500? 4,500? Oh, okay. Is that deep? Yeah, it's gonna get really something like 4 degrees.
Actually, what?
Huh.
That's why we use it for
Pretty negligible.
All right. Okay. Stop.
Stop.
That was 750.
We'll help move, please.
Okay.
Correct.
875.
Go over there.
Me? Just checking. No, I wanted to do like a wanted to take photos, I think it, in the microscope, using the bioluminescence technique.
Okay.
You know? Could be like-
I'm sorry, was it? Okay. It was something. Good choose.
Oh, I see. You can share. I didn't. You're sharing.
So what I was doing was I
Uh-huh.
was trying to find photographs that
Uh-huh.
you guys collected
Yeah.
on some of the specimens.
Yeah.
So like on the live stream, there's just a couple.
Oh yeah.
photos
It's
going
funny.
through.
I didn't even see that till now.
Yeah,
Oh, cool.
I just did it.
Awesome.
And then, so like some of the bioluminescence you
Oh,
guys
cool.
were trying to take photos of.
Yeah, that's awesome. That's
Yeah.
like one of our most exciting results.
Yeah, it's super exciting. Yeah.
So postdoc Lee Miller, who is working with our team at Mbari, is doing this super exploratory work to see if we can quantify how much of this marine snow is bioluminescent, because there's a lot of kind of anecdotal evidence, just people who have dive, like people who have done deep sea diving, or in people who go in like submersibles, see all this bioluminescence, and obviously that comes from animals, but people say it's also the marine snow, and there's a couple papers out there that quantify it, but almost nothing. So Lee set up this camera to do really long exposures of our samples of the marine snow, which we collect in this really special, like, delicate way. And surprisingly, like, first try, saw all this bioluminescence. So we're really excited. Getting close. Nice. All right, 875.
Question for you. Are
Yeah.
you going off on that one or on mine?
Where is it on yours? Oh, I see.
Because I'm
Yeah, that's fine.
I'm using this
That's
one
fine.
as a base.
Great.
And we
Yeah.
are a meter and a
Yeah,
half off.
it's really
It's
fine.
okay for you.
Yeah. All right. Are we good to start? Or you have to catch up with the ship?
Oh, yeah, you just go get started and I'll start him in a second. And then we'll be fine. Alright. Ready?
Yeah.
Bridge RV, continue that move, please.
Okay.
Thank you.
All right, we ready to start?
Yes.
Okay, I'm starting.
All the way.
Starting. This is 875. Okay. Where are those Phaeodarians? I was promised.
Too late at night.
Yeah, whenever you want to find them, that's when they hide.
How do your sediment traps work?
How do they work?
Yeah. How do they collect? 'Cause
Um,
they're just like cylinders.
well, they have a bottom.
Yeah.
It's like an open cup, basically.
And you just let it kind of like drop down into it.
Yeah, so then they're out there for 3 days this time, and then so it's just whatever falls in over 3 days. Whoa,
Yeah.
cool. Is how it collects. So it's just kind of passive sinking. And then some of the tubes, the collection tubes we have, just have you know, salty brine with a preservative on the bottom, so that so the stuff doesn't keep degrading after it's been collected.
Yeah.
And then we can measure, you know, how much carbon is in there. So it's just a chemical measurement, or we use it for DNA and get all the DNA that's in there. But then the other thing we do is we have a gel layer at the bottom, so the particles like just fall into that gel layer, and they stay separated from each other, so we can put them under the microscope and image them and really see what they are and how big they are, and like what type of fecal pellets they are, or what kind of animal they might have come from.
So this is just like more of a visual of each layer.
This
And and
for
we're
the
not
ROV?
necessarily- Yeah, and we're not necessarily collecting anything from the ROV.
we are getting the animals will be quantified from the ROV. I would love to figure out a way
Like
to
a net, like a little mesh net that they tow behind them.
Oh, I mean the imagery. Yeah.
Isn't
This
it?
is like the imagery is going to quantify the animals. Just we'll use the video to count and identify how many animals were at these depths.
Who's gonna get stuck with that challenge?
What's that?
Who's gonna get stuck with it?
Well, I mean, I hope that it's actually not a person. I hope that we can just use some of the machine learning tools that, you know, like my colleagues at Ambari have been spending decades developing. So I feel like that should be a doable task, and that the real human work will be after the machine makes its best prediction. A human will need to go through and actually correct it, because it won't be you know, all accurate, right? So that's where maybe Anila's team, you know, they're the taxonomy experts, and Tracy, who's not on the cruise, but they're not really experts in the image processing. I'd say that's more my team is kind of, that's our wheelhouse, but then the taxonomy is their wheelhouse. So
They're so small.
yeah,
All these particles.
the particles, yeah, I would love, oh yeah, so many larvations at this depth. I would love to figure out a way to quantify.
I was just curious with how your sediment traps do it, because I was just like,
The
I feel
sediment
like
traps-
that's such an easy thing to, I guess, adapt to the ROV, which is.
Oh well, I mean,
I don't know like how much you would catch.
well,
moving
the
forward.
sediment traps need to be moving through the water as passively as possible, not moving. So there's some sediment traps that are just on like neutrally
Or we
buoyant
just
floats.
go like forward, and it goes in. The angle.
Well, the point of the sediment traps is to collect what's sinking, not just everything that's there, right?
I want everything.
I mean, we could collect well, I could ask you to collect individual particles with the ROV.
Well, that's what I was thinking more of, because like these particles would be more like
They're
what
so
we do with nets, right? Like
yeah,
the fine mesh nets.
right. I mean, we have collected like like the abandoned larvation houses. Those are big enough to collect, but they're really delicate. But like surveys or when you take a camera and put it through the water, this is really good at getting how much particles are in the water. And so like we're doing that with our camera on the CTV, the ISIS camera. And that doesn't tell us how much is sinking, but it does tell us what's in the water and how much. But the sediment trap actually specifically targets what's sinking. Where are we at? Let's see. 9 more minutes. Ooh, there's something really cool and dark lower there. Ooh, nice one.
Hey, Ghost. Hey, Ghost. Everyone just sit there at the top. You're in the sweet spot on the head.
Ooh, nice.
Yeah, going to 15 on transit.
Ooh, nice. Okay. Ready for dance party? All right. That's the end. Stop.
Please draw a view.
It's
Another stop, please.
Copy coming close.
Exactly going to now?
1,000 meters. Ooh, big larvation. I'm probably gonna finish the transects, and then Anitra's gonna show up for the fun part.
Same with us.
Yeah.
There's been cruises that I've been on where every time we're in the water, the other shift will launch us and we'll come in and we'll just fly through the entirety of our 12 hours.
Uh-huh.
And they'll come on and leave us and then not to come right away for
And-
like weeks at a time.
Yeah.
They don't.
And they never get to
They just
fly.
Good job. Cool. So what animal do we have to see during the transect every day? Just stop.
Oh, maybe like a lanternfish.
Okay.
Any crazy, like, really black fish.
Seen a few of those.
Yeah. On the transects?
No.
Yeah. Um,
Better than most, like we'll be doing these things in the seabed, and they're like, okay, this is gonna be a transect, we're not gonna stop, turn, this is this kilometer away. And I'm like,
uh-huh.
Lost like 30 seconds.
Yeah.
Like, what is that? Like, nope,
Yeah.
no.
Yeah.
Can you pull up a camera of the docking head? Just put it anywhere. The docking head. just so I can see what the envelope is doing on the surface.
Which one do you want?
The camera for the docking head, it's showing the docking head.
Okay.
Yeah. You can put it anywhere other than like the currents. The starboard hanger maybe something, I don't know what it be.
This one?
No, the one that's sticking out the door.
This one?
Yeah, and you bring that camera up so I can see the back of it. Can you tilt it up?
Oh yeah. Just let you know.
Keep going. That sucks. Alright, I can't see it. Never mind. It's stuff in the way for some reason.
You want to see the wire?
Yeah, I want to make sure that the docking head is in a position where it's not affecting the wire. I need to be able to see it like reference to the coming out from the bottom of the docking head.
Mm.
Is that any better?
Yeah, it's pretty good. I'm probably smart. Thank you. That's 1,000 meters.
All right. Let me know when you're ready.
Let's see. Can you just like move again for me, please?
Great, you can continue the move.
Cover that computer.
Put on the.
That's fine. No, you don't need to keep it on. I just wanted to look at it.
I don't know if he has that. I think he broke that.
How's that? Speed wise? A little faster?
It can be between 0.2 and 0.3.
Alright, push it just a touch. How's that?
That's great.
Alright.
Okay, I'll start it. And target depth is 1,000. Great. Now,
Would you rather me go off of that one or that one?
what's that?
Depth wise, would you rather go up that one or
Oh, I mean, what, they're off by,
a meter?
I mean, and also
Meter and
where's the camera at versus the CTD versus that sensor. It's all kind of like, you can go off of whatever you want to.
Alright.
A meter, to me, it doesn't make a difference. Because, the height of the ROV
Yeah.
is, I don't know,
Yeah, the depth sensor
it's
is
within the uncertainty there.
here, and the CTD is.
Yeah, exactly. So, if what we're quantifying at one meter off is different from the meter above, then we have, observation statistical problems here. Ooh. Excuse me. What is that? Looked like a larvation house.
If you could come a little bit to your starboard, I'm getting blown to the northeast and I can't really come back to you.
Yeah, no problem.
Thank you.
Cool. Mm. Searching for those Phaeodarians. Ooh. Sometimes there's tricks. There's ones that are not Phaeodarians. Oh, wow.
Mhm.
Is that a 4M?
Mhm.
Beautiful. That was huge. Or it looked big.
That was about the same, yeah. It's about the size I've seen.
Yeah.
Yeah.
Ooh. Hmm. Beautiful larvation there. It's interesting, we don't see a lot of the, well, now that I say that, it's funny, I was going to say we don't see a lot of the abandoned larvation houses except right now. We just flew by one. But
I see a lot of whale sharks. Maybe you talking to big existence. You talk to
yeah.
empty
Oh
ocean.
yeah, now we're going to see one.
Yeah, it's the hope.
Yeah.
So much better.
Oh yeah, someone's asking about what's the difference between forams and radiolarians. Forams and radiolarians are both part of this big super group of protists called Rhizaria. And so they all had a common ancestor at one point, and then as they evolved, they diverged, and one group split off. That's the forams, and all of these are single-celled organisms, so just a single cell. And forams form this calcareous shell, and that's what we saw. We saw it was kind of glowing orangish in the middle, or it reflected a lot of light. But then they have these like it's the cytoplasm part of their cell. I think it's called, I don't remember the name of these kind of stringy, it's basically cytoplasm that they extrude out from the cells, and that's how they collect tiny particles on that and then pull it into the cell to digest it. And so those are made out of calcium carbonate, and then their distant relatives, the radiolarians, are also single-celled organisms and make shells out of silica or glass, and they're incredibly ornate. And then the other sister group within that it's also related to radiolarians is the phaeodarians, which we saw quite a few of those yesterday or whenever our last dive was. And those are also single cells, but often in the deep sea here they grow in colonies, and so we can see them on the ROV, and those are the ones that kind of look like Anitra thought they looked like COVID-19, but I think they're too beautiful to associate with such a sad virus. But they also form silica structures, and it's just incredible because they're single-celled organisms, but they're really large. We can see them with our eyes, and they also are so complex. They form these mineral shells but they're just are really beautiful, and it's just a single cell doing that. Oh, can we tell the difference between radiolarians? Is that what I think that's what they're asking? Can we tell the difference at a glance between a radiolarian and a foraminifera? Yeah, definitely. At least I can. And definitely in the ROV, they look pretty different. If you look at them under a microscope, they look, yeah, they're totally different. And the things that are made out of glass, the silica, they just refract light in a really different way than calcium carbonate. Can you tell what was the name of the third type of rhizaria I mentioned? Is the phaeodarians. They used to be called radiolarians, but then as people did more work on them and DNA and all sorts of stuff, we realized that they are just outside of the family group of radiolarians. Oh, nice. Wrong camera. So yeah, they definitely have been called radiolarians, but now their term for them is phaeodarians with a PH. And actually not very much is known about them. And my group has started studying those organisms in the last few years. Oh, that's it. All right.
Bridge RV will pause that move, please.
At 1,000. Now we're going to 1250. Ooh, another sinking larvation house. That's funny. As soon as I was like, I'm not seeing any sinking larvation houses, and then we start seeing them. Yeah, that's how you spell phaeodarian.
Josh, you gonna join us for our culinary tour in the posters? It might not happen actually. It might not happen.
Yeah, if you're out there, it's coming back. I think they're another good.
You're up on 2026? Good guys.
Well, we might not have one because reasons. But I'm holding out hope until all my hopes are dashed. Maybe out with it.
Like it's old or not? That might have been one of those field, whatever.
You saw it?
Something just came up that looked like it was in a later stage of development.
Oh.
It had that same
The same shape?
shape and translucent.
I didn't see it.
Tagged by something that's stuck on me.
Hmm. I see it.
It's
Mm.
got, I don't know.
I think maybe it's a remnant from that big red one that we ran into. Ooh. All right. Oh wait, we're going to 1250. Okay. Excuse me. Oi. Jeez. The sneeze were heard around the internet.
Yeah.
So, Colleen, your sediment traps only go down to 500 meters?
Yeah. Mm-hmm.
And we're going down here to 1,500 meters due
Right.
to the other, I guess, fishing we've been doing with the mock
Yes.
nests.
Yes.
And, okay.
Alright,
Okay, 1250.
Okay.
Let me know when you're ready.
Great, you could continue with that one.
Sorry.
Alright.
Okay. Start.
Work.
Fine.
Wait, start.
Where do you wanna go?
Here we go. Start. Start.
I think this is yeah.
So
Okay.
you wanted to act up at the exact wrong time, so it's a timeout for the rest of the night. How is it? How'd you sleep?
We went to 1500.
Mm-hmm. All right.
What's up, Michael?
Morning.
How are you today?
Very well.
Right now we're at, it's in at 12:50.
It's gone, we're just creeping down or staying.
We're staying at 12:50 for this one, and then we're gonna go to 1500 for the next one, and you're gonna come up with like wonderful.
Oh, we're still doing the fun better.
Yeah. And right now you don't have anything on other than cruise and heading, so no trims. Actually fairly
Good one.
pretty well trimmed. You'll have fun.
Okay.
It's really kind of cool. Have fun.
Okay, good nest. Happy with speed?
Yeah, we've been kind of going around this between 0.2 and 0.3 has been good in this current. So. Oh.
Oh, you've started
Yeah.
this one already, okay.
What's that?
You've started this one.
Yes, it started. It's going.
Alright, we're going down to 1500 again.
Yeah, so there's only one more depth after this, or 1500 is the next depth,
And then the ones that are,
and then the exploration starts.
no,
That's probably about when I'm going to bed.
Oh.
Whenever Anitra comes to relieve me.
Yeah.
Yeah.
So how's it been looking?
Good. There hasn't been a whole lot. Like, I wasn't here yesterday or during the last dive, so I didn't see everything, but I saw the previous station. Like, there's way less jellies in the water here.
Oh, okay.
Yeah. But the video looks good, and the ROV it looks like it hasn't been doing a lot of pitching today, which has been really good.
The current is a bit less here, is it?
0.6? 0.5?
Oh, really? It's just something about this location compared to the previous. It's just,
Yeah.
it looks so much better.
If the currents are really high, like the other day, the current
Yeah.
was really high.
Yeah.
The floats and everything are kind of pulling your back.
Yeah.
You're powering
Oh,
into the.
that was it.
That'll
That's why.
probably be
Yep.
it's just kind of
Exactly. So,
focusing a little bit.
yeah, that makes sense. Okay, so that's what was happening before. Oh, I bet that's like one of those eel snout things. Sorry, no stopping.
There's a few things down here.
Yeah.
So far.
There's been siphonophores. There was a depth with quite a few larvations. Wow.
There's a long one.
Yeah. That's probably the biggest one we've seen actually today anyway.
that's really good.
That's a big one. Wow. definitely way less stuff down here in the water. I mean, even the particles, just like way less particles.
Yeah. What's been the most active zone on the way down so far? This dive.
Oh, let's see my notes. I mean, so for collecting on the way up, well, it's going to be different too.
Yeah,
with the time, right?
Yeah. But if it all stays in like darkness, like should it be relatively similar, or?
I don't know when they start. Anitra knows. She's been keeping an eye on the timing of when they come down, when they swim down, and it's really fast. It's crazy how fast they swim down. I think it's something like 7 in the morning, or maybe earlier, 6 or 7.
The sunrise starts at like 4 a.m. Just now.
Oh, okay. Well, maybe that's when it is.
Okay.
Okay.
It's been getting light really early.
Because definitely in the other night dives, they've kind of, by the time they get back up to the surface, the animals are like down.
Okay. Yeah.
But let's see, in the 125 meters, we saw a lot of these, what we think are larvae of, like eel larvae. These flat, like clear, wormy things.
Nice.
Like they don't have appendages coming out. It looks like a ribbon.
Okay.
Anyway, so there was a ton of those. Then there was more squid up there, but obviously, I guess because it's nighttime. There was more larvations at like 607, 625, 750. That's when we started seeing more larvations. And I wrote down ctenophores at 875. So.
Good spread.
Yeah. We haven't seen like a giant amount of fish. Kind of some random ones. I guess they're all at the surface right now.
Usually there's quite a lot of larger things at this depth.
Yeah, there haven't been that much big things actually, except for that giant siphonophore we just saw.
Show up as well.
Good folks.
But you could go a little you can go between like 0.2 and 0.3 in terms of speed.
The speed shouldn't really have changed, but I can bump up a little bit.
Yeah, because we should just be going like it should be coming constantly, like through. We should be going through the particles constantly, basically.
The current's got a little bit stronger.
Yeah, that's better.
I it's pumping too much. Yeah. quite like it.
That was cool fish. I don't know what it was. Maybe somebody in the chat knows.
What if we alternatively did one run where we shut all the lights off and see if we saw bioluminescence? Just go straight without the lights.
Yeah, that'd be cool. I think you might need lower light cameras though to do that. Right? Have you ever seen bioluminescence with these cameras?
Yeah.
Oh, you have?
Well, when we were in the middle of a krill storm,
Oh.
you had to switch the lights off because the krill was too much. You couldn't see for the krill anywhere.
Yeah, yeah.
So when we turned all the lights off to
Oh,
let
cool.
them leave, you kind of just saw these things like
Oh, wow.
pinging past.
Oh, that's neat. That'd be neat. Yeah, my colleague at Mbari has a images bioluminescence in the deep sea. And Steve Haddock, and he has like a special camera that does low light imaging, so it's like these super beautiful
That'd be awesome.
videos.
Should've brought one along with
Oh,
you.
no.
Next time.
We'll get Steve to come next time.
Yeah, that would be cool. To have
Yeah.
a proper dedicated camera for it would be really cool. How much longer have we got in our time?
Surprise. I'm not going to tell you. You don't have much longer to wait though.
Oh, I say 1 minute 30 seconds.
Alright, we'll find out.
What's your bet?
That's more.
What?
I think you're going to be both pleasantly surprised.
How much?
More.
4 minutes. No. Alright.
Alright. Stop.
Okay,
That
stop.
was the 1250, so now we just have the 1500. 1250.
And down. Shit a little bit. Okay, let's stop it. Look at that. There we go.
Okay,
Where are you?
good.
You ready to go?
Sure, yeah. You came just in time for the last transect.
Oh, okay.
And then you get to do the exploring.
Alright. I'm ready to explore.
All right. All right, I wrote some notes on like what we saw at different depths here.
Oh, you did?
Yeah, I mean, it wasn't like there was a depth where it was super concentrated. Maybe, you know, the shallowest had the most slurry of life in it, but
Do you have any requests for how much time I spend at different?
I mean, you have so many hours, so it's like go crazy.
I guess I could divide it into intervals. And
Yeah, I mean, you might as well spend some time at these deep depths, right?
I guess so, yeah.
You could see some crazy stuff. Maybe you'll see a anglerfish or something.
Dive down to 2,000 meters if you wanted to.
Yeah, could. Okay. Well,
I'm supposed to stick above the 1,500 mark though, I think.
oh, I don't know.
We're diving down to 15 now.
Yeah.
All right, so I got to start the event.
Yes.
You're using an iPad?
Yeah. Do you know how to do it?
Mm-hmm.
It's really simple.
Yeah, I have it also on my computer.
Okay. Well, you can do it however you want.
Okay. Oh, that takes a long time to get down there and bring these transects.
Oh. Yeah. Okay.
I did a lot of things while you were doing that.
Oh, good. Like fun things.
I mean, I hung out in the lab for a long time.
Okay.
And I went and laid down for a few minutes.
Oh,
I
good.
just had a cereal party with Lamella
Oh, good.
and Alon and
Yeah.
Ronnie.
Okay. All right. Well, I'm gonna go get some sleep.
Okay.
Good night.
And we will come whenever he comes. I
Yeah.
assume.
All right. Good night.
Good night.
I'm glad you found our photos. Because
That's great.
we've been uploading them all on the shared. Cool. Yeah. I'm glad you like the latest photos.
I love it. My face.
Cool. So, ROV's coming on at 8? Is that the plan?
Yeah.
Okay. And then we
That's
go transit to the,
Tier 7 tracks.
yeah. How far away, where are
We're
they?
not that far away.
24 nautical miles or something.
Only 4 miles.
How far away are we from the stop?
Yeah.
Last time I checked, probably about 4 hours ago, it was 15 nautical miles away.
So it's like less than an hour, or no, that's about an hour.
2 hours.
2 hours.
Oh, it's a 2 hour, okay.
Yeah. So it might be 2.5, 3 hours by the time we get back.
Okay.
To go from here to
Yeah.
there, okay.
I thought it was closer to 24 nautical miles on the screen. It's there.
They're 24 miles away now.
I think so.
Well, just like when I
I'm
should
just
be
in my
prepared
bed,
to-
so I don't know. Don't take anything I say
But it sounds
in
like
gospel.
it's not gonna
I've
be
just
at
finished my
eight.
first coffee of the day.
They're getting on deck at 8.
Yeah, could be like 10. Alright.
Alright, we break. Do you want me to come to pause if you're done with levels there?
Well, I'll be ready.
You can slow it down just a little bit. I'm managing to keep up with you for the most part. Just drop the speed back and we should be
Let's
fine.
maybe do another mock nest tomorrow night.
Got me coming at point 2, more like.
That would mean we think we'll get your stuff. in
Morning.
time to do that.
Oh, I think we will.
It's like you might be at your first one coming in, have it in by 1, go to the other one by 2, and then by 3, that's it by 5, 6. We should be okay.
Yeah, I think hopefully we'll be faster than that, but that's a good. I think we're
And then we can come back.
great.
But we could mock nest back too.
So probably what would be best, since the RV's coming up, at some point move the mock nest out of the way to help with recovery, is before I think we moved it over so people have more room for the recovery part, because that's what we're
Of
going to be
this,
doing.
of the traps.
Of the traps, yeah.
What
Okay, we probably want to put it
I
back
don't know
in
where it
tomorrow
is.
night.
Yeah, I mean, I don't know where it is. I didn't.
Like all we did
We
was
were-
move the mock nest over, like it was still on the half deck, we just
Yeah.
lifted it up and
Yeah.
moved it over. So we had more
Like
room for the
forward
recovery.
or-
No, to
Starboard?
Starboard, yeah.
I defer to your judgment. Yeah. Whatever
Because
you
it's not
think
gonna
is
take,
best.
I don't think it's gonna take like a ton of time to move the mock nest back.
If you give the mock nest a little shove, is it gonna like, are wires gonna pop out of it?
No.
Or, okay.
No.
I know.
It was just like,
I know.
Maybe just don't touch it.
I know.
Had everything to do with the bottle on the inside. But
Yeah.
Okay, I think whatever you think is best,
Yeah.
if I win.
I think at the end of the day, what's best is having a clear deck for recovery of the buoys.
Okay.
That's just me. But,
Yep.
I will do whatever.
well, it's not, it's just, it's not
Yeah,
that, it's
I-
just, I'm just thinking.
I agree.
She's
But,
there.
yeah.
And if we're gonna do the mock test afterwards, just me.
Oh, we're almost there. Okay.
Yeah.
All right. You can say night night to Colleen. She's leaving now, and I'm taking over. My name's Anitra Ingalls, and I'm a professor at University of Washington.
Yeah.
All right. Night night, Colleen. Have we seen it? I guess you guys haven't had a chance to really see anything exciting because you're moving.
There was some stuff at the last depth. It looked pretty cool.
Alright.
A few things have gone past.
There's a jelly, dinner plate jelly. What would wow me? Oh boy, what would wow me? Yeah. Oh, where's this? Are there things you guys hope to see when you do these deep midwater dives?
Everyone's just a surprise. Sometimes you see loads of cool stuff, other times you maybe hardly see anything.
Right. And
I
this,
think everyone wants to see, I don't know, squid are pretty cool if they
Look-
behave and want to stay still for the camera.
right,
They're
right.
not darting about all over the place, but jellyfish can be pretty cool as well.
Right.
I think everyone just wants to see something and get some nice close-ups and
Yeah.
cool shots.
Yeah. I was really enjoying the really small things, and it's kind of amazing. You can see them so well. But I study single-celled organisms, so I get excited about the things like forams that are single-celled that you can see, the protists. But that's maybe not what most people get excited about. That I haven't known.
So hard to see.
I haven't. Yeah, I know the ones that I saw yesterday were the Phaeodarians, and Colleen's been telling me about them, and they're really very interesting protists, and I would like to see some more of those today. The ROV has lights, yes, and we're illuminating the water around it. It does scare some things away, so we don't see those things, but not everything.
The university that wants to be seeing all the big stuff, all the colossal squid.
The
and
colossal squid, yes. I mean, that, well,
the
that would be.
whales and stuff. They're just like, nah, not going anywhere near you guys.
No, yeah.
Right.
Are we there? Oh, yeah, we are. Okay,
Yeah,
so
yeah.
should I start the event?
We get the speed and the depth just right.
Okay.
Pretty much on it there if you want to start.
Okay. All right. Video transect. Target depth 1,500 meters. And okay. Start. There we go. I started it. The radiolarians are pretty awesome. I like the phaeodarians also have silica shells, and they're closely related to radiolaria. They used to be in that group. I guess they've been moved out to a different group. That's what Colleen was telling me. But this is 1,500 meters. This is as deep as we'll go. We are also using nets to trawl for animals and collect them, and our nets go down to 1,500 meters. So we're also doing the ROVs to 1,500 meters. They're asking how powerful the ROV camera is. I don't know exactly how to
Powerful.
by that. Powerful.
Yeah.
I mean, those phaeodarians are, I mean, maybe like a grain of sand size, you know, less than a millimeter, and we were zooming in on those pretty well.
Yeah. I mean, it's a 4K format that we've got.
Yeah, but the zoom in, I guess, like how much can it magnify, right? That's maybe the question.
Official figure.
I don't
Quite
know.
a lot, but we would look for more in our
Yeah.
next camera that we get.
Oh yeah.
Because it's never enough. You can
Never
get so
enough.
Close,
I
close,
know.
close, close,
I
close.
know.
Jog that with aye, it's running that. You can, 1.1 1.2 1.3 1.4 1.5  1.6 1.7 1.8 1.9  1.10  1.11  1.12  1.13  1.14  1.15  1.16  1.17  1.18  1.19  1.20  1.21   1.22 1.23 1.24 1.25  1.26 1.27 1.28 1.29 1.30 1.31 1.32 1.33 1.34 1.35 1.36 1.37 1.38 1.39 1.40 1.41 1.42 1.43 1.44 1.45 1.46 1.47 1.48 1.49 1.50 1.51 1.52 1.53 1.54 1.55 1.56 1.57 1.58 1.59 1.60 1.61 1.62 1.63 1.64 1.65 1.66 1.67 1.68 1.69 1.70 1.71 1.72 1.73 1.74 1.75 1.76 1.77 1.78 1.79 1.80 1.81 1.82 1.83 1.84 1.85 1.86 1.87 1.88 1.89
Ooh. Siphonophore, yep. That was a good one. We're not stopping. We got 10 more minutes.
We're ready.
You're ready.
Fine.
You guys have to take bribes to who gets to have to do the transect. Who doesn't have to do the transect?
I stepped into the wrong seat this morning. I was like, oh, I thought we were already on the way up.
Yeah.
I like doing the Midwater stuff.
Yeah. Yeah, but it's nice when you can explore a little.
You can actually stop and see loads of stuff.
Yeah.
I would be like, oh, I would stop for that.
I know. That looked like a sea butterfly or something.
It'll be fun. At least we've came in at the right time where it's like the end of the transects, and now we can do more fun stuff.
Yeah.
Hopefully get some cool stuff.
I mean, we bring these, we trawl with the nets, and there's nothing we can do but have them exposed to the pressure change as they come up. And some of the animals do okay, some not so much, but that's the only way we can really sample them. There are some people that try to keep things under pressure. We have a colleague that has pressure tanks, and you can bring the animals up and then quickly put them in a pressure tank, and they can then do experiments on them. But we are not doing that on this cruise. We probably do it on our next cruise. I think it'd be hard to catch a fish in the deep ocean in a pressure, like in a tank that you could keep pressurized, just because it's hard enough to collect them with the ROV samplers.
Yeah. Fish are fast.
So we rely on the nets.
I mean, we catch fish with nets as well, with the ROV. It's one of the fun things.
Oh, you have a net?
Handheld.
Really?
Yeah.
Well, I don't think we know about that.
Yeah, well, it's
maybe we'll get that out for their next time.
a kind of difficult thing to do midwater, maybe, because they are fast. But
okay,
it's more creeping up, and you need to kind of be able to
shrimp.
You kind of be able to get the slow moving fish near the seafloor.
Right. Yeah.
But yeah, we've got a good few fish before.
Mm-hmm.
It's challenging though.
Mm-hmm.
But like a challenge.
Right. It's good. I mean, these fish do swim up and down on their own. They experience that pressure change every time they migrate up and down. So it's not something they've never had to do before. Watching.
Can you tell watching as well? Okay. Yeah, it's on there, yeah.
Four. No, just joking.
I was thinking.
150 days.
Oh, okay. All right.
4,600 total through the whole dive. So people that came on and
So far with this one dive?
yeah, so there's
Wow.
160 watching at the moment, but
Right.
then 4,500 over since we started have came and gone.
Wow. Wow. That's about the attention span nowadays, I would say.
Yeah,
It's like a good, it's a long reel. That would be a long reel.
A lot of the stuff on YouTube that I would watch would have to be like that to cut off for me.
How come this is like in this? Oh, there. Okay, yeah, just up there. Gotten below 3 degrees Celsius. It's chilly down there. The oxygen's not too low, 50%, 53%. I'm a lot more used to working in the Pacific where the oxygen gets a lot lower.
Does it change like at the same time of day as well? Like the oxygen zones, do they fluctuate with the migration of the animals, or?
You know, I don't know. I think not. I don't think the oxygen changes that much in the deep ocean. In the surface it will because of photosynthesis. But I don't think it changes that much on a daily cycle. That's a good question. Maybe it does. I mean, there's always a lot of respiration happening because the microorganisms are doing that all the time.
Yeah.
And I don't know how much that animals add to that. It's a good question. Yeah. I think it's probably pretty minimal. But like in the Pacific, it'll get down to 2, 3, 4 micromolar.
Yeah.
In a huge part of the Pacific.
Yeah, we've been areas on the way out to Rapa Nui that was like really low oxygen levels, but there was like hardly anything in the water as you went down. Like it was like nothing. You could go the whole descent. And usually you'll see something, like a few things on the way down. But I mean, there was countless dives down there where it was like there's like hardly anything in the water at all, not even particulate or anything in the water.
Huh. Really?
Yeah.
Interesting.
And then you go other places and it's like, whoa.
Right.
Can't even see.
Yeah, I mean, here there's a fair bit of particles here, and a lot of them are swimming around, meaning they're a lot and there's something alive. There's a shrimp. Copepod. Yeah, I mean, people. Yeah, it's interesting. That's the jellyfish. To me, there's a lot more particles than I would expect in these deep waters, but it's hard still for me to get a sense of the scale of like what volume of water are we looking at. You know what I mean? Like,
Mhm.
is that 1 liter of water or 100 liters of water we're looking at,
Yeah,
or more?
takes some working out.
Yeah, and they have to work that out when they do the video
Yeah.
analysis of the video. Okay, so our timer went off.
Oh.
We
Okay.
are ready to stop the event. Video transect, stop. 1500.
Okay, Bridge, can we just come and I'll stop in that move just now?
Yeah, I'm gonna stop.
All right, that was our last depth, survey depth.
I can change light settings and stuff like that if I want now if we're done with all that.
Well, whatever settings you think are best for kind of looking at things, and
Yeah, I like a few settings. Aren't there?
it's brighter.
My question though is when we're doing these video transects, how can you better quantify what you're seeing as far as like, cause like in the sense that with the ROV we know exactly what our depth is,
Mm-hmm.
but when we do nets, a lot of it's kind of when we're towing it, it's guessing, and then we can reprocess where the net was at a certain time period with a depth sensor that's logging internally. So I was just wondering, like, would it, like in the future, if you were to do something similar, would it be beneficial to try to figure out, like, a way to catch the marine snow to see if there's differences in the marine snow as far as like when you're talking about like smaller particles? Would that be like an interesting thing to like try to quantify, or?
Yeah, I mean, there's a whole lot of people that do attempt to do that in different ways. There's you would not use nets to catch the particles because they're too small.
Would be like I guess a certain type of mesh would be.
I mean, I don't think there's so there are filters that people can send over the side of the ship and filter water in situ. So there are people who do that, and actually on your next cruise, any of you are going to be here, you will see Hillary Close doing that. She's bringing a set of in situ pumps, so they have a battery usually and a filter holder and a pump that pumps water through a filter actually in the water, and you can put them down as far as you can put a CTD, I guess, you know, and then you can filter tens, hundreds, sometimes thousands of liters of water depending on what kind of filter you're using.
Yeah.
So some people use a 0.2 micron filter and try to catch all the particles and the bacteria as well, and then some people use like a glass fiber filter that is a 0.7 micron mesh, which lets a lot of bacteria through, but it collects a lot of the particles. So that's almost like it's sort of like a net, I guess. But it's you have to force the water through a filter that size. You can't just let it flow with the natural currents. So yeah, that's one thing people do, and then.
But then I guess another way is to like fire like the niskin bottles and then
Yeah.
kind of sample.
Yeah, you can collect the water, bring it on the ship, and filter it. And you'll get different answers if you do those two different things, partly because you're filtering such a different volume of water, and there's just error associated with that. And the way that the pumps bring water through is different than the way the CTD catches it, right? So
That's why it's important to have scientific processes.
Yeah, well, yeah, we do the best we can.
You want to constrain, I guess.
The CTD is a conductivity temperature depth sensor that goes down with a rosette of bottles called niskin bottles, and we can collect water with that. And it also measures conductivity, temperature, and depth. And then you can put other sensors. We have fluorometers, we have transmissometers that measure particle density, we have oxygen sensors, we have pH, nitrate, cameras. We put cameras on ours. to look at the particles. So there's all kinds of things you can put onto the package to put different sensors there. And then, I mean, Colleen is collecting the sinking particles in sediment traps, so that's the other way to, if you want to measure the things that are sinking. But that's a different pool of carbon that you're collecting when you're collecting things that sink. But there's also something called a snow catcher. They send this 100-liter volume to, it's like your jelly jars, but it holds 100 liters of water, I think, 100 liters, maybe more. And they just, it's like, it closes like a niskin bottle, and then they bring that up. So they get a really, and then they can watch this marine snow settle in it, and they can sample it at different depths in that column. So there's different things people have, you know, invented to try to do this.
Yeah.
That one's called a snow catcher. Dave Carl has one, and University of Hawaii that he uses. But then there's also like chemical tracers of particles. So particles absorb thorium, which is a decay product of uranium. So uranium is soluble in seawater and just stays in the water, but when it decays to thorium, it absorbs onto particles because thorium is particle reactive. And you can measure the amount of particles in the water from the thorium deficit. So there's just also these sort of chemical ways of doing it. Oh, we've been getting a lot of those in the nets, I think. Yeah, oh, that's pretty.
Focus is the train with the it's harder to see the things when this trays out. I think you don't want to catch this.
No, I don't think so. I'm not sure how many things we'll catch or want to catch. We'll see, I guess. But you could catch it if you want. If you want to practice your skills. If we see a Phaedarian, I'm going to want to catch the Phaedarian.
Underneath us. Not just floating through the gap.
I don't know much about Phaedarians. I've only really just learned about them in the last couple of days from seeing them in the ROV and I showed a picture of one to Colleen. She wasn't here when we first saw it, and she's like, "Oh, that's a Phaedarian." She was all excited. They are protists, single cell. They secrete this sort of silica, I don't know, skeleton that's very delicate, and they have, I think, other cells that kind of attach to the outside of this sort of sac, and hopefully we'll see some. The one we saw looked kind of like a coronavirus, but bigger. We see a lot of the Phaedarians in the sediment traps, so they're sinking, and so it's possible they're important for carbon flux, and there's been some suggestion that they are kind of important for carbon sinking.
says it is there, but should be still be looking just in an I've got a lot. Yeah, okay. It's got a lot.
Ooh.
But look at this.
Oh, sure.
They're going
I can't tell what it is. That might have been a larva. That might be one of the Phaedarians. It looks too big for that, though. Oh, maybe it is. Maybe that's a maybe. I'm actually a chemist, so I'm going to do my best at naming some of these things. That might be a Phaedarian. That looks kind of like a Phaedarian right there. Yeah, that's what it is. Pretty sure. Or it's a. I don't think it's a foram. I think that's a Phaedarian. You can tell it has that sort of not perfect looking sphere, and then the spines coming out of it that are silica. And then the bright dots might be additional cells that have attached to it, but I'm not 100% sure. But that is a Phaedarian, and I would have not been able to identify that a day ago, but thanks to Colleen, I'm able to. Wow, cool. That's kind of awesome.
Do I want that?
What? Yeah, we could try to collect that. If you could collect that, Colleen would be excited to have that. We might do some like genome sequencing on it or something if it comes back intact. It's crazy that they can make these very delicate spines. People study these silicifying organisms because they lay down very pure silicon dioxide, which is kind of like what's in your silicone chips. And so there's a lot of material scientists that study this biological silicification process. Oh, there he goes into the, God, close him in there, yay! Oh my God, we got it, Phaedarian, woohoo!
That's it.
I think that's more like a millimeter or less, or maybe it's bigger. I don't know. How big was it? It looked kind of big. I think they're not that big, but maybe they're bigger than I think. Some of the single-celled organisms can get pretty big. I don't know.
Tiny. You can just kind of see him there, I think. Can you?
Oh yeah, he's like a millimeter. The silica shells are amazing. The diatoms also have them, and the radiolaria and the Phaedarians. They have the organisms that secrete silica they make special organic compounds to precipitate the silica out of the seawater, and those compounds are referred to as polyamines and silifins, which are peptides or proteins.
If I zoom out, it's right behind that ram. See it just in the edge, but if I zoom
Is that another one? Or did it get out? Oh no, he's in there.
It's like
Oh
right
no.
behind the ram there. I'm just sort of peeking.
Can you see it in there?
Yeah, just
All
it's right behind the ram, but
Okay,
it's kind
all
of
right.
hard to.
We'll see if it survives the journey up.
Yeah.
They think they're fairly delicate, but
Okay. So.
I used to actually study the organic molecules that are used to precipitate silica, so it was a big interest of mine for a while. Get my pointer out.
Yes.
There's a shrimp.
Gotcha. Shrimps would be hard to catch.
Yeah, very hard to catch a shrimp. We have a lot of shrimps in our nets, and they come up pretty happy. They're usually do just fine with the pressure change. We saw a shrimp today, actually, in the net that had this blue fluorescent or bioluminescent tentacles, and a person who's our crustacean expert had never seen that before, so that was kind of amazing. Wow. Cool.
He's fast.
Ooh, there's some nice focus. Wow.
You can try and get them if you really want, but they're pretty quick.
No, I don't think we need to catch a shrimp because we have a lot of those.
Yeah, let's just keep going then.
Yeah, but it was fun to watch it swim there for a while. Like there's a maybe, I don't know.
So my focus on 2 seconds. Well, see this or no? I just think it's a
That's a larvation, or is that a?
larvation.
Yeah, that's okay. I mean, we can, we don't have to avoid it, but we don't have to spend too much time. But if we get closer, it's probably going to explode.
Oh yeah.
Oh, they tend to kind of, yeah. That one's actually holding up.
Do I do that always? So again, Leo. No, it's gone. It's dispersed.
Yeah, those are the houses of the larvations. Oh, this thing. What's that?
This thing?
Yeah, down there. Yeah, that thing.
Yeah.
I can't tell. It looks like it's swimming. Maybe. Oh, yeah. Oh, wow. It's like a swimming blob.
Coming right at me.
Yeah.
I think it's just dispersed as well.
Yes.
Oh, there it is, maybe.
I don't know what it is. We can keep going.
I don't know, whatever it is, it's moving around.
Yeah.
Fast.
Yeah.
Keep going.
Yeah.
Excuse me.
Shrimp.
Amazing.
There's something.
See that? Yeah, see
It
that?
went off to the side.
saw it.
Is it too late?
Maybe.
Might be some sort of jelly. Oh, there he is.
Well, again, it's kind of caught in our thruster wash a little bit,
A
but
little bit, yeah. That's okay.
probably again. I do the ISO again, Leo.
Oh yeah, the evening lighting. Wow. cool. I have no idea what kind of jellyfish that is. So, what time is it? It is, we have 7 hours, so we could.
See this thing? It's quite slow.
Is that a siphonophore?
Again, wheel.
Hmm?
Sorry, it's just to get a wheel. Sort that. Exposure. Tiny little siphonophore.
Oh, that's a little siphonophore, yep.
What, bump up one? Yeah. Yeah, it's better.
That's kind of cool, those little red lobes getting twisted up. Wow, they're like sparkling. Sort of incredible.
Focus doesn't like me to do it though. A little bit more tricky with this. All this stuff out in the bottom, it's harder to see them. Champagne.
Start.
Okay.
I guess we could slowly be coming up a little bit as you go. Oh, are you leaving?
Yeah, we're doing an hour.
Alright.
Hour around.
It's like lifeguards, you know.
Yeah.
They take like 15-minute shifts, right?
Yeah.
Oh,
Yeah, 15 minutes to see 15.
Okay. Oh, come on.
Take that.
How are you?
Good. Yeah.
You good? I think if we slowly come, up in depth as we look, that could be good because eventually we'll need to get to the top. We have about 7 hours, right? Yeah.
Okay. Coming up slowly. See if you find something on the way.
What is your name?
Leonardo.
Leonardo.
Like Da Vinci.
Right. Do you go by Leonardo or Leo?
Leo.
Leo. Okay.
Yeah.
There's 2 Leos.
Actually, in Portuguese is Leo.
Leo. Okay. Are you from Brazil?
Yes.
Okay.
From Brazil.
You're going
From
home
Rio.
soon. Oh, you're from Rio.
Yeah.
Wow. You gonna show
Going
these
back
guys
home.
around when you get there?
Yeah, I'll try to show some good places. Some good food.
Yeah.
Have you been to Brazil?
Never.
No.
No.
Need to go to the steakhouse if you like.
Uh-huh. We did that in Argentina.
Yeah. You compete with them. Yeah. And go to the Christ Redeemer.
Uh-huh.
It's a
Yep.
very good sightseeing over there.
Right, yeah.
The Sugarloaf. Really, Rio is really pretty. It's a mess, but it's pretty. Too crowded.
Is it?
Yeah. Too many people.
Yeah. We are in the UTC -3 time zone. Yeah, it's 1 o'clock now, almost 1 o'clock in the morning. 0100. There's a larvation maybe, a larvation house there maybe, and a and there's a siphonophore next to it. The siphonophore is poking the larvation. I don't think I've seen that happen yet. Wow, that's quite a. I'm surprised it's staying without disintegrating. There it goes. Yeah, they're so delicate. There's a lot of people that study that snot in the ocean. That's what we call dissolved organic carbon. There's a lot of it in the ocean. Look at that. Wow. Cute. There's the, is that a Keating gnat straight ahead? That might be a Keating gnat, yeah. If it is, it'll probably jump away soon. Yeah, I think that's a Keating gnat. Oh. Huh. Maybe not. Maybe that's it. I think that's what it is. Oh, no. Is.
Better?
Yes. Yeah. Seems like it's just floating around. If that was a Keating gnat, I would have, that was alive, I would have thought it would swim away by now. I don't know what that is. Arrowworm, yeah. It's just like it just seemed like it was asleep for a little while there and woke up. Very weird. Mm-hmm. you become an ROV pilot?
So I'm an electronic technician, and that was my first job.
And was what
After
kind of
I-
technician?
Electronics.
Electronics. Oh.
Yeah. And I left the technician school and started ROVs when I was 19. So I've been doing this for
Oh wow.
24 years.
Oh wow.
More than half of my life.
Yeah. Wow. What is that? How long have you been with SOI?
7 months.
7 months. Oh wow.
Yeah.
Where were you?
Before I was in oil and gas.
Okay. Everybody seems to have come from there.
Yeah, that's the most.
Lost it.
The market for ROVs, the most is on oil and gas.
Yeah, that's what I've learned. Another cubic math. Another larvation. What in the world? Wow. That's just wow. Wow, just poof. Now that's part of the dissolved organic carbon pool.
Mm-hmm. It's gonna become food.
It's food for the bacteria, and then the bacteria get eaten by protists and things like that, I guess, and then yeah. Got you in here now. What is the whose pictures are showing on that one screen? Are those Alex's pictures that are scrolling through on that screen?
They're just the pictures that Josh took from the scientists to display. So if you want to describe them, I can put them up on the.
Oh, Josh took them?
No, they're from you guys. I think.
We took them? Like Science Party took
Yeah.
those? Oh. And who on the Science Party? Do you know? They look like Colleen, a lot of Colleen's pictures, maybe. Are they showing on the, can the audience see those?
1 second.
I thought I saw one flash. Yeah, there, okay. I think those are these are the sediment traps. Those are collecting things that are sinking, and that's one of our net trawls right there. That looks like a fecal pellet, although it could be something else. You're going by very fast. Ooh, Bella swimmers, wow. Now the particles all seem to be swimming around suddenly.
Yeah, because I'm coming up.
Mm-hmm.
Maybe that's.
Is it better to go at a constant depth for a little while and then move up? Do you think? Are we more likely to see stuff if we go straight?
Sometimes just by chance, you come up and see and go horizontal and see. But we can spend some time the same here, and then forward, and then come up some
more. Oh, that's a heating math. I wonder, like, what some of these little particles look like. Are they just alive, or are they? There's a little tiny. That's probably a copepod up there. That's a copepod. maybe just check out that one. I don't know if it's anything.
Focus, try to.
Yeah. Hard to focus that close up, huh?
Yeah.
The little tiny things. It's okay.
Kind
I
of
was just
spikes.
curious. Yeah, let's keep it like this maybe for now. What's that? Maybe it's just a particle. I don't know. Yeah. make it go up a little bit, maybe. Ooh. Those are pretty. They want to know if we'll see it if we turn out all the lights. I don't know if we want to do that. is. I guess that's a larvation. think we've seen one of those. Maybe. I guess that's the same as the other one, yeah.
It's similar to the
Yeah. Kind of shiny. Should come up a little bit more.
coming in.
Yes, yeah, you're slowly coming up. I can see that. I see a lot of. Can we see the ADC pan here? Or we don't have it on, or do we?
Yeah.
Sorry?
Is the ADCP on?
Yeah.
Can we see it? The data?
Where do you want this? Where do you want this? It's up here. Show you somewhere else. I'll put it over there for you.
Usually it's the one with the arrows showing us which direction the current's going, right? Yes, that's a different display that I'm used to seeing. That's currents with depth. Last time we got up, the surface of the currents were so strong around 500 meters that we couldn't really do anything, so I'm just kind of curious.
Here.
Oh yeah, that, okay. So, that's pretty low. Start getting higher, up 500, yeah, it's kind of good there. Alright, well, we go up a little bit to like, I can see in this acoustics that there's not much animals below about 1,000, or at least that are reflecting on that.
You want us to come up?
Just slowly. You can, yeah, sure. Yeah, slowly though.
Okay.
is. Oh, there's a little Joey.
Baby
Wow.
Stop swimming. Very small.
Mm-hmm. Whoa. Hmm.
Come on now, guys.
Ian always brings the chocolate. Oh, look at the tentacles on that guy. Ooh, wow. Wow. Whoa. Huh. It's like it's such a weird thing. Another one flying a little too close to the sun there. Coralia. Elevation. Gonna go away. I'm seeing a comment about cotton candy, and that's something that if you take the dissolved organic carbon out of seawater and you dry it down, it looks a lot like cotton candy, and that's because there's a lot of carbohydrates, so it sort of looks like cotton candy. That's one of the things people call that. There's a keating nap. Maybe we keep going up. Sometimes there's a lot down at depth, but maybe not at night as much because they're up higher.
Yeah. There's seaweed in here.
Mm-hmm.
Close to the surface.
Mm-hmm. Oh, wow. We're so lowly coming up a little higher because, yeah, we're going to look for things a little higher up in the water. And we have until about 8 a.m. our time. On some of our dives, we've seen quite a lot of larger, pretty interesting organisms at this depth, but it's a little slow tonight, so maybe we'll go up a little higher and see what we see.
Treating guards.
Okay. Wait.
Okay.
You guys switched seats. I don't remember your name.
I'm Andy.
Andy. Okay. Hi, Andy. Nice to see you. How are you doing tonight?
Doing good.
Excellent.
Staying awake, making it.
Oh, Ian brought this.
Some sugar.
Some chocolate. It's helping me stay awake.
Coming upstairs here, it's not something that
We're just coming and seeing what's there.
maybe stopping at specific depths or just stopping at specific depths or just
We're just kind of slowly going up. I think we were getting a little. Things were a little slow, and so we thought, well, let's go up a little higher. I think their audience is talking a little bit about the you know, whether things are in the deep water during the nighttime, and it's true that a lot of things do swim up to the surface to feed at night, and that's one of the things we're studying. So I still kind of expected to see a few more things down here, but we're going to go up. Maybe I can see in the acoustics that maybe around 1,000 meters things start to get a little bit busier, and so maybe we'll go up a little bit above 1,000 meters and kind of see what it looks like. I'm sure there's a ton of things at the surface, but there's also stronger currents up there, so it makes it really hard to get a very good look at anything. And so the currents are a little lighter down deep, and that's why we're spending a little bit of time down here. Ooh, some fish. Would you like some chocolate?
No, thank you.
Can you pass it to Ian?
Of course.
Get it out of my face.
Thank you, Ian.
No worries. Very important.
On the ship here, the chef doesn't really make dessert. Once a week we get ice cream. On Sundays we get ice cream. And otherwise there's chocolate bars and a few other sweet treats. So I have this idea that the crew has asked them not to make dessert because, you know, maybe they don't want dessert every day of their life, or maybe twice a day they don't want dessert. But you would eat dessert every day.
Religious crew mandated.
Yeah, it's definitely. They don't listen to us.
Have you requested desserts? No.
Although last cruise they were cookies.
Yeah. I've been on other ships where there's dessert served twice a day and snacks in between, all home baked. It's a little bit much.
Oh yeah.
Where are you from, Andy?
I'm from California.
Oh. What part?
I grew up in the Bay Area, right between San Francisco and San Jose. It's called Redwood City.
Okay. And did you start in oil and gas?
Yeah, I started out in commercial diving.
Commercial?
Yeah, commercial diving. Saturation diving. So I was a technician. Used to take care of all the diving systems.
And did you do a lot of diving yourself?
When I was in dive school, yeah. I used to dive all the time. After I went to work, that pretty much stopped.
After what?
After I went to work, that pretty much stopped.
Oh, I see. Mm-hmm.
There wasn't a whole lot of good scuba diving in Louisiana.
Is that where you did the oil and gas stuff?
Yeah, down in the Gulf, yeah.
Mm-hmm.
Got our ROVs. Yeah, okay. Uh,
Well, that's one of those, jelly things. We've been catching a lot of those. We caught a fish that had these kind of a thing in its stomach. Been eating it. Yeah. Ooh, very pretty. Current doesn't seem to get too bad until about 500.
Yeah. Santa Barbara.
Oh wow.
Yeah, a lot of fun.
What kind of sites?
We would load up this hunk of junk Corona we had, and just drive up the coast and look for kelp beds.
Oh.
And pull off and then try to figure out a way to get down.
Mm-hmm.
Swim out, dive kelp.
I've always kind of wanted to swim around kelp some more. What kind of things do you see in kelp forests?
Oh, on the coast there in California?
Yeah.
Oh, you got the Garibaldi's, sheep's head, down at the bottom, you find the little ledges, cutters, like the little anemones.
Mm-hmm.
So, of course, you got the little, called the pistol shrimp.
Mm.
No, it's popping, like pop, pop, pop.
Yeah, I can hear them.
Yeah.
Mm-hmm.
Once in a while, you can see some halibut in the bottom.
Oh wow.
Sand.
Mm-hmm. Salmon?
No.
I guess there's not many salmon left down that way.
Not that far south.
Mm-hmm. Or maybe they're, I don't know how far south they used to go, but.
And then every once in a while, that was the cheap dives, right? Because it was just gas back then. That was gas cheap.
Mm-hmm. Mm-hmm.
Getting out to the Channel Islands was a bit more.
Let's see. Hmm.
Much clearer water.
Mm-hmm. Yeah, I bet. I haven't gone diving around Seattle at all, or coast or anything. We kind of let our, I'm certified, but I kind of let it lapse.
That's like riding a bike, you forget.
What's that? Yeah, no, I know, but you lose your certification and then you're not technically supposed to.
Huh? When did they start doing that?
Well, like if you haven't gone diving, if you don't dive like once a year or something, then they kind of want you to go take some more training.
Really?
Patty does, yeah.
Yeah, I got certified when I was like 11 or 12.
Yeah.
I kept doing it.
Mm.
High school,
Yeah.
dive school, and all that stopped.
Mm-hmm.
Something straight ahead? Yeah. Up a little guy? I think the only diving I've done in the past 15 years has been to clean the bottom of the boat.
Do you have your own boat?
Yeah.
What kind?
Back then I had an Islander, monohull, sailboat,
Oh,
and now I had a catamaran, 34-foot catamaran sailboat.
34 foot?
Yeah.
We just went down to Australia. I've never been before. And we rented a catamaran sailing boat with some friends and
Nice.
sailed around the Whit Sundays for a week. And that was pretty fun.
Yeah, that. Yeah, that's right.
I never really spent time on a catamaran. It was fun. I liked it.
Yeah, it's very different than a monohull, right?
Mm-hmm. It was good because there were two families and we each had our own hull.
Yeah. Yeah.
And the kitchen in the middle.
A lot more room, right?
Yeah, a lot of room. They couldn't believe how much room there was. They were used to sailing in a monohull, so.
Yeah, it's a different deal. This cat's pretty nice. They fit a lot in 34 feet.
Mm-hmm.
It's got like a queen size master forward and 2 cabins aft.
Mm-hmm.
Nice big U shape down there. Yeah.
How often do you get out on it?
Well, not as often as I'd like to. I don't live. I have it in San Francisco Bay. And currently I live up kind of in the mountains.
Mm.
So I'm like 3 hours from the boat.
Oh, wow.
Yeah. I just moved to cameraman because the things are quick there. This summer I want to take the boat down the coast though.
Oh, yeah.
Go back down to Santa Barbara area, Channel Islands.
Mm-hmm.
And just
Just that up there is the fish.
Oh, yeah. Oh, he's gone.
Now they swim away. Which jar is that has that? We collected a. Ooh, there's a ctenophore, yeah. Wow, that one's really glowing. Are those jars labeled from numbered from left to right?
Yeah.
We put a Phaeodarian in the number 3. I didn't log that event though. Probably should have, right? When I collected the something.
Yeah.
Maybe I will. Maybe I'll do it, even though it's late. Okay. Biology sample.
Wait.
Good answer.
Yeah.
When we did that jelly jar one,
No me.
stand up.
I still know here.
I didn't look at it, no, I was flying at me. I was getting doing other things. This is what the
I've made my point. For the purposes. Yes, I recorded.
I guess I logged it a little late.
Yes, please.
Is that a dinner plate? Is that a dinner plate jelly?
that was somasis.
Now we're getting into where there's some more stuff.
Just seems now. First video.
Did you do much sailing up there around Seattle?
What?
Did you do much sailing up there around Seattle?
No, I don't. I'm not much of a sailor, but I have friends who are. My son was sailing for a while, sailing camps and stuff, but we live right next to Golden Gardens, Shilshole Marina, but a lot of sailing goes on there.
Something sausage like. Yeah, lots of that. Some sausage like feature.
Are you hungry, man?
Well,
Someone said bless you as I sneezed. Thank you. Whoa, that's a weird thing. I don't know what that is. Oh, we saw one of those yesterday, actually. We did see one of those. It was just looked a little different.
Zoom in. Zoom more. It's nice and stationary. Zoom, zoom, zoom. Lovely waterfall. You're getting mistaken for a water moustache.
It is like a caterpillar. Are you controlling just one camera, or are you controlling each?
You only have control over one. This one just kind of goes up and down.
Oh, okay.
This one will handle tilt all up.
I see.
This one's the one with the most control.
And who's controlling this one?
Mine.
Okay. Oh my God. guess you have to count the segments, but that guy's so clear.
Zoom in a little bit more.
Wow. It's like a real, wow. That's the first, like, sort of crustacean thing that's hung around long enough to really look at it. It looks like its head got chopped off, but I don't think it did. Wow. How big is that thing? Wow, that's pretty big.
You're missing a jar.
Oh,
I mean, jar number 2 is best.
Jar number.
you don't want it, yeah.
Do I want it? I mean, he's pretty cute, but I don't know. I don't think we need it. But
Fair enough.
if you want to practice your skills, you can. We can always let it go. Pretty cool.
There it goes. Yeah, he's- Oh, man. Stuck.
There's definitely more things here at 1,000 meters than there were down at 1,500.
Yep.
But sometimes we were seeing a lot of stuff down deep. I guess not at night. Ooh. Fish. Have you gone into the lab and seen any of the trawls that we brought up?
No.
You should check it out. Tony came in.
Oh, yeah.
Mm-hmm.
Yeah.
Hmm?
When's next?
Well, tomorrow, we're hoping tomorrow night. But if the weather chases us away, then we won't do it.
See it.
That green thing. We're at 1,000 now.
It is.
Oh, that's a. Is that a Oh my God, that was a hatchet fish, I think. Oh, that's so funny. It was just sitting there. I'm learning a lot about these different organisms. I can really do better with like diatoms at identifying things, little phytoplankton, but now I have to learn all the animals. Oh, hello, Delano. Are people still in the lab? What? Were there any more big fish?
No, not yet. They're processing the net 2.
Net 2?
Net 3.
Net 3. Okay. 2:30.
It's not in the form of cooking. You know that's the trick,
On here? Not yet. Not yet. Do you want me to? We have one Phaedarian. It's a protist. We collected it. Well, Colleen, when I showed Colleen the picture of it yesterday, she said, "Oh, did you collect it?" And I said, "No." And she said, "Oh, you should." So I did. But we have still 5 other jars to fill. So if you want to sit here and tell us to pick something, we'll do it. Or if you want me to collect a jelly, I will.
Jellyfish will do.
Any jellyfish?
Yeah.
Even if it's tiny? Or do you want a bigger one?
No.
Any jellyfish. Okay.
Yeah,
There's one right there. Yeah, okay.
If I find any students who process all the samples,
Yeah.
then do some genomics for that.
Yeah.
From Brazil, yeah.
Each one of our trawls usually has a few hatchet fish in it, little tiny ones. We just saw one. That's it.
Today we sampled a different species. Today we sampled a different species from the other hatchet fish that we got the other day.
Okay. Yeah, we saw this transparent, I don't know, maybe it was a euphausid or something. I don't know what it was. This thing. It's like totally transparent. There's a little tiny jelly. I don't know, is that...
Is that glass right? No.
No, maybe. Maybe.
Very beautiful.
I don't know. I was like,
Tiny, tiny.
do you want to get that one? I think that's a jellyfish. Maybe. Yeah, maybe not. Yeah. Maybe it's a tinophore.
It's really tiny.
Yep.
Yeah.
Yeah, I guess it's a tinophore. It's sparkling,
Yeah,
right?
it seems like it's a conifer. A beautiful one.
Mm-hmm.
Conifer and the Unidaras are related somehow, so.
Mm-hmm. Do you want that?
That's a jelly.
You want to try it? That's a dinner. Is that one of? That's not the dinner plates. That's a different. Oh, it's tiny.
It's really tiny. Maybe find out a bigger one.
Yeah.
One of those things there. Orange thing with that floaty bit at the top.
The floaty bit.
That's the center. That's the Latin name. Parachute. catch it or no?
You want to catch it, Delana? Do you want that? I don't want it.
Maybe it's too tiny. I don't know.
Yeah, it's small. I don't.
It is. It's like, yeah,
This is
tip
like
of the,
I don't
it's too
know.
tiny.
I can't quite tell like what its story is, where it's, you know.
Maybe find it.
No.
Yeah. Make it pray. What kind of eggs do you put in this?
Scrambled.
Scrambled. What was it yesterday?
Yesterday was the
Egg loaf. Both.
Egg loaf? Oh, I can do no more. Yeah. Like 3.5 weeks.
Maybe we should come up a little bit shallower.
Coming up.
I don't know, not too much, but
We can go up. We can go down.
oh, we
Just kinda-
oh right, we can go back down. Why not? Oh my God, it's late.
Did you sleep today?
I mean, I thought I I mean, I slept last night and then I took a nap. So I thought I'd be pretty awake, but it's, it's late. So, I don't know. I've been sleeping such.
I think it's like only yesterday.
What?
The day before yesterday, I think I slept. Almost. I don't know. It kind of lost, the sense what day you are because
Yeah.
we're just sleeping these crazy time.
I know.
I slept today from midday to from 1 to 5,
Mm-hmm.
and that's it.
Right.
Yeah.
Well, I went, I think, after breakfast and slept till lunch. So I thought it'd be easier to stay up tonight, but I got kind of tired there for a little while.
I'm exhausted because all the work and I've been doing.
Mm-hmm. Here's a little jelly. You want it?
It's so beautiful. I think this is even smaller, no? Yeah, he's pretty small.
Are you hoping for a jelly that fills the whole jelly jar? Substantial one. Yeah, might in there.
Don't need to be that big, but
No.
a little bit bigger than that.
Yeah, that first dive, we had that giant jellyfish, didn't we?
Oh, yeah, okay.
You want a rice with jelly?
Jellyfish.
What is that? It's a worm, right? Oh my god. No, this one would really capture my jellyfish. They don't agree with that. Is it a giant copepod? Hmm?
It seems permanent. You have to get back to the center. When you find like a jelly bigger than that one, then you want to do some. Good catch. Nice.
Bigger than the micro jellyfish.
The micro jellyfish.
Okay. Oh my god. Stand up for me.
Or there's so many of those.
Oh no, I'm not
Giant.
one chocolate.
Sure.
Just, yeah.
Chocolate
Stretch my legs. When have you guys heard anything about when we have to get out of here from the storm? It's coming.
Friday?
It's Friday, yeah, but I'm wondering if there's anything more specific.
Not at all. I'm not sure.
It's a forecast. So it's the forecast.
Yeah. Yeah. Well, I didn't know whether Jason wanted to get a head start on it by, starting before it gets here. Like it's moving fast towards us or something.
Is it? You have not looked up the weather recently, but I can look up the
I
weather.
think it probably plays into what
It's out of it.
No, I think we have to get out of here one way or another. I mean, we won't be able to do anything. That's what he said.
Yeah.
Oh, look, just up.
Where?
Come straight up from where we are.
Coming up. That?
Oh, right there now. No, just something. Maybe it was that. I just saw something. Sausage shack.
Sausage links.
What he is. The guy.
I'm going up to these new hunting grounds. Searching for giant jelly. Those are the jelly. They're looking for giant jellies.
Giant jellies. Ones that barely fit in the jar.
They don't have to be that big. Hello. Hola. You do anything fun on your time off?
Nothing. No, I just caught up on some emails. Now I'm excited for some hot seat action.
Yes. We could probably be slowly ascending.
Yeah. Let's do some snot. We're not interested in that, are we?
I don't know what it is. Our Portuguese-speaking scientist from Brazil just left the room. He was here a second ago. this wispy organic stuff.
This, maybe? I don't know what that is.
That long thing,
Yeah.
that's a arrowworm, kitty gnat.
So that's it. What was looking at? This is away from too close, I think. Yeah.
Yeah, the kitty gnats kind of tend to run away.
Larvations, see the larvations always look really good from far away.
Mm-hmm.
You always catch out.
I mean, they are cool. There are a lot of them, so you can't look at every one.
Hunt for some good jellyfish. Yes or no? No.
Mm-mm.
Playing in the winch, yeah. Interested in these? Raveluk or no?
No. We had a bunch of those. There's a larvation.
That's a big one.
Yeah. Cubic math.
That's just 950. It's the same. Same thing again, I think. Same one.
That's another cubic math, yeah.
It's a larvation. See from miles away. Yeah. There we go. Oh, it's a little jellyfish or something.
Mm-hmm.
Do you what ISO for me, Leo?
Mud. Let's take a look.
ISO.
Let's take a look at it.
Just bump up a couple. Yeah. I don't know. We'll just get
Wow,
some
look at
views.
that. That's kind of amazing. We could collect. Is that a jellyfish?
I don't know, is it?
What?
I guess it zooms a bit first anyway.
I guess it's a jellyfish. It's pretty interesting looking.
You want to collect?
I guess not. I don't know. I mean, that's kind of looks like might be hard to do now. Maybe that's the kind of thing he wants, though.
It's there, if you want it.
Okay, sure.
What do you want it? What jar do you want it in?
Anyone. The only one that's got anything in is 3.
Can get it in 4, maybe?
Sure.
It's really flowing. He's in there.
All right, we got one. That's for
Got it.
That's for Delano.
So do we need to note that?
I'll log that. Yeah. I'm I'm awake still. Kinda. That was 4,
Yeah, that was 4, yeah.
and that was a jellyfish.
Look at this, whatever it is. Maybe a fish. Try.
It might be another arrow worm. I think it's a maybe it's a fish. That's a fish. I doubt you can get it.
I'll get images of it, but we won't probably.
Wow, look at him, he's just hanging out.
Do I take? Leave the ISO down just a touch maybe. See if it's still within the stream, and I'll try and get a little bit closer. There we go. Oh, focus, come on. He's going. Oh, focus doesn't like me. Yeah, look at that if you want. Looks like there might be a jellyfish or a solstice behind it as well. Do you want to look at this?
Sure.
Do the auto ISO for me again, mate? Sorry. The squid.
That's a squid? Oh yeah, wow. Look at that. That's a cool squid, isn't it? We would take the squid maybe. You don't get a lot of squid, but wow, look at that.
All focus, so.
Wow. I think it might be better not to take it because it's going to take a lot of work to process that. Everyone's exhausted. I mean, it's not I'm not kidding. Take the squid. The audience wants us to take the squid.
Squid will be hard, but you can try if you want. Do you?
I don't think I
Two.
do. Oh my God, he's going in there. Nah, he's tiny. Let's not.
No.
No. No. He's too tiny. It's really cute though.
So I'm just gonna go in himself.
He's trying to go in there.
Do you want that or no? You don't want
No.
it? Okay.
Even
Trying to get better at shots when but not all focus is really not liking them. As well.
I don't know if I logged where I took that jelly from. That was 950. Number 4. Oh my God.
Stop coming so close to me. electric flight into the camera. Coming up. Elevation, is it? Yeah.
Mm-hmm.
Same things we've seen. Not interested in that. I take it.
No, that's another siphonophore.
Yeah. This guy. Same. It's just a cypher.
That's. Oops.
Got to look at her and you're not worried.
If you want to look at it, we can look at it. I think that's another siphonophore.
Du har också från den. Ja.
Hmm?
Touch the deal.
Uh-huh.
Dice up a little bit, mate.
Hmm?
Sorry, we go again. Set. Too close. One's getting blown right towards us as well.
Yeah. Maybe we come up a little bit more.
Yeah.
Ooh, what was that?
Wow, look at that.
Is that a jelly?
Some Tino.
Cuneiform? Oh.
Look at her or not?
No, it's going.
Not really.
Hmm?
Not really. No.
No.
Yeah.
We already saw a couple of those.
And some big misses.
Is that a dinner plate jelly? I don't know that he wants that kind. They're so flat. Maybe he does. I guess you could try to collect that one. It's a jelly. I'll get a picture for him.
I'll bump up the ISO one.
Yikes, wow.
You want that?
Sure.
Yeah, for sure. And then 5.
Log it. Okay. Biology sample. Why are you laughing at me?
Because you're like,
Jellyfish.
Okay.
Number 5. Oh, I'm already going to put it in there. Okay, I think it's a. Somebody said that might be a.
That's
So.
so misses, yeah.
Storage location, jelly jar 5. Submit. Okay. That was, we're at 9:19. I'm just a little tired.
Yeah.
I'm really fighting
I
it.
get some good sleep last night, no?
I mean, I did, but
The days catch up with you.
it's late.
Yeah.
I mean, I even had a nap this morning after breakfast, but
Yeah. The days at sea catch up with you after a while.
yeah. We've just been on really irregular schedules with everything. It's just
Yeah, I bet.
crazy. One day to the next, it's just completely different.
That's cool. It looks pretty weird.
Well, I don't know what. I was going to say it's another one of the siphonophores, but it is not. What is that? Oh my, wow, that's a fish of some sort, huh? Wow.
So maybe one more. Yeah.
You almost could catch that one, but let's not. Wow, pretty cool.
Try and come around that a little bit, maybe a better view. Got away from me.
It's like, what are you doing here? Wow, what a crazy body form that one has. It's just like with all those tentacles and the long tail.
It's really low as well.
Somebody changed the font size. I can't really see the comments anymore.
That was me. I need to do a reader.
What?
I have to read a direct stream every 8 hours. So.
Oh, what is that?
Just changing something.
What's it do?
This is betraying me a little bit though.